Okay, hello everyone. My name is Amanda Rosenzweig. I am the biology department chair and part of the SLT program. One of the concentrations is in biology. I wanted to go over a few housekeeping rules for today. Whoops, there we go. In your toolbar on Zoom, you can choose to select the chat and introduce yourself into the chat room, and that way you can talk to the other individuals on the line. Please remain on mute if you are not speaking, but feel free to raise your hand, and when we call on you, you can ask a question. We do have a few minutes reserved for each speaker at the end for questions. And then we also have live closed captioning that's available during the session. If you want the closed captioning, you will click on the CC icon in the toolbar. During this, feel free to tag us on social media. Use the hashtag DCCSLT, and you can find us on social media through Facebook, Instagram, and LinkedIn, and at DCCSLT or at DCC_SLT are our tags, our login information. And then there's also all of the contact information below for both biotechnology and chemical technology, as well as the individuals in charge of the programs. And it is my honor to introduce two individuals. One is Ms. Charlene Schunig. She is the biotechnology director. And the other is April Noble, Dr. Noble, and she is the chemical technology director. But today in April's spot, Deandra DeMole is going to speak on chemical technology just to get us started. So I'm going to hand it over to Ms. Schunig, and Charlie, I'm going to stop sharing so you can share. Thank y'all, everyone. And again, please feel free to put anything in the chat. In addition, I have put the links to our handouts today in the chat, so you can grab them to save them to your computer.
So before I begin, I just wanted to check with Deandra if you wanted to go first. If not, I'll just begin.
Sure, not a problem. I can go first. Okay, so Delgado offers an Associate of Applied Science and Laboratory Technology. And so I want to stress that a lot of people are not aware of an Associate of Applied Science. And what that truly means is that these are going to be courses where you're not just sitting in a lecture, you're going to be actively involved in your learning. This is going to be active learning, hands-on. Not only will you learn the knowledge, but you will also gain the skills that are needed for employment. I'm going to focus on the chemical technology part, but the overall program is a limited admissions program. So we look for students who are truly engaged and willing to be successful in this 2-year program. In the past, most chemical technicians were pretty much trained on the job. So for example, in laboratories, they're taught to do titrations. And so you can be taught to do a titration. You're just pretty much opening a drip, almost like a faucet. But if you don't understand the chemistry behind it, then you'll never be able to advance in your career. And so what we want to do is we want to not only train you with the skills, we want to make sure that you have the knowledge background for that. As moving forward in the future, these jobs are going to demand a solid foundation in applied basic chemistry and mathematics so that you can do calculations, so that you can measure the quality of your performance on the job. Quality assurance is a job that you can move up to after you've been a chemical technician for so many years. They also expect you to have some amount of computer knowledge and oral and written communication skills. And we also make sure that you have those types of skills in this program. We do things like mock interviews, and we help you with resume writing. We want to start you in this program, but the finished product is you gaining employment upon completion. And that is our goal. The best way to prepare for these jobs is a 2-year degree from a 2-year college. And Delgado has the advantage of being the only community college in the state to offer this. And why that is important for gaining your education in the state is because most of our students already live here. You have already set your roots here. You have a family, and so you want to gain your education here and then go on to get your employment. The state of Louisiana hires approximately 3,000 chemical technicians. 1,800 of those are in the Baton Rouge and New Orleans area. The average wage for this position is $65,000. Now, that is a lot of money in the scheme of 2-year degrees. I have 6 years worth of college. And let me just say that that is not my base salary. So these are well-paying careers. You're familiar with this region, you know that there are a lot of oil and gas refineries, and they hire a lot of chemical technicians. And as I mentioned previously, these were people who were just trained on the jobs. What we see happening now is a lot of these positions, the people are retiring. So they were grandfathered in, but now they're retiring out. And so there's going to be hiring of a lot of new, younger laboratory technicians. Also back in the day, they would promote lab technicians to chemists without them having an actual full 4-year degree. But if that's something that you choose to do, we can also show you how to transfer to a 4-year very, very easily. So once you leave out and you go into this position of a chemical technician with the 2-year degree, you don't have to stop there. You can continue to be successful in your career and move on to higher levels. So we know that the field is changing. We need more chemical technicians and also biotechnicians. Although biotechnology is not my area of expertise, as I'm a chemist, I know that that field is growing exponentially, and there is a dire need. When you think about jobs when you're a kid, or even when you get to high school, a lot of times we know, I want to be a fireman, I want to be a doctor. These are the jobs that pay good salaries that people don't mention. I mean, no one grows up and says, I want to be a chemical technician. But now that we're grown, we understand that, hey, there are jobs out there that I know that I could work and be successful at, and it will be fun. When you get into the laboratory and you start producing things on your own and you can troubleshoot and figure things out for yourself, that is the joy of actually being in science. And that is what makes applied science much better than just having a general knowledge degree. When I finished my bachelor's degree, which was a chemistry pre-med, I knew all of the book material. And my first job was actually a chemical technician. I worked in a lab as a contract employer. The company was called Chemtech, but it was contracted by Freeport-McMoran. I'm not sure if you're aware. And so we tested a lot of metal ore samples. Freeport did a lot of drilling back in probably the 60s, 70s, and 80s. So I knew all of the textbook knowledge, and that's where they used me for the most part, but I didn't have the actual skills. And so I know that that is missing, and that is what is needed for chemical technicians. If anybody has any questions, feel free to ask me any questions. But that is all I have for chemical technology.
Thank you, Deandra. Next up, we're going to have Ms. Shunick speak about the biotechnology. And I am going to mute myself and hand it over to you, Charlie.
Okay, thank you. So I'm going to go ahead and screen share with y'all on our website and kind of introduce you to how to navigate to even locate more information about our program. And then I'll introduce you to our curriculum and all the different courses that we have to offer. Can y'all see my Delgado homepage? Okay. So I get this question all the time, especially when we do go to classrooms and students are here in our program. One of the biggest issues is our program has a really long name, Science Laboratory Technology Program. And most of us refer to it as SLT, but then underneath that we have two concentrations, biotechnology and chemical technology. So usually what I say is go to the search bar on the Delgado homepage and type in Science Lab Tech. And that's the most efficient way to go to our information webpage within the Delgado website. So once we open this up, it's going to bring you to an overview where you can just learn about even what Science Lab Tech is. And then you can go and get more information about our concentrations. And this is where you can find applications, information about the curriculum, contact information, all that kind of stuff.
Charlie, I'm sorry to interrupt you. We can't see the actual webpage. It's only your
Okay.
desktop.
That is what I was worried about. It should be this one. Can you see it now?
Yes.
Okay. So yeah, let me go back then. We're going to start here once you click on where our web page is, and then you can go and start navigating through our concentrations. A lot of students ask if they need to pick a concentration right away, and the answer is no. And I've found in the past that some students actually want to get skill sets for biotechnology and chemical technology, depending on what their interests are and what kind of career and field they want to go into. Some people are strictly biology, some people are strictly chemistry, but for example, if you were interested in forensic science, you would really want to study some of both, what each concentration has to offer. So I am the biotechnology director, so I figured I would go over some of our curriculum and kind of talk about the skill sets that we give you as briefly as possible. And what I find is what students typically do before they even apply to the program is take our intro lecture and lab, that's Science 130 and 132. And at first, students know, I think I want to be in a lab space, you know, this and that. Once you've taken those intro courses, you get a much better idea about which path you want to go. And then we can kind of tailor your experience to, you know, your end goal. Do you want to go on to a university? Do you want to go straight into the field? And what type of field do you really want to study in? So here's where you get info on biotech. And here's where you get info on chemtech or chemical tech. They're really similar fields, but of course we emphasize different things that you're going to be working with. You know, in biotech, you're working with cells and tissues and bacteria and things like that. And in chemical technology, you're working more with chemicals. I mean, I think that's probably more obvious, but here's where you find our application. And this is where you see the minimal requirements in order to apply. And this is accurate for both the biotech and chemical tech pathways. And usually what I recommend for people that are still on the kind of fence about whether or not they want to join the program is go ahead and enroll at least in the intro lecture, where you can get all the background on the different fields and what you can do in a lab tech career. And then this is where our course plan is. So it really aligns pretty closely with people wanting to go into healthcare fields. And oftentimes we find a lot of students that find out they're not passionate about maybe nursing or medicine, but they are still passionate about working with people and treating disorders and diseases and all that type of stuff. So a lot of these classes may be classes that students already have to take or already have taken, depending on where you are. But it is a lot of biologies and chemistries and a couple maths, which is probably not so unexpected. You start with Gen Bio, Gen Chem, college algebra, English composition. As Dr. DeMol mentioned, having good public speaking skills and good writing skills is extremely beneficial in a career path for lab tech, because you actually do a lot of that in your career, especially as you keep moving up and up in your job. And if you do go to a university, you know, you probably will need to do some interviewing and work with instructors and professors doing research. As you continue to move through, it starts to get a little bit more specialized. You'll take your intro classes, you'll do some microbiology on our end. We start doing a little bit more chemistry. I find that some of us, because I'm one of those people, we can be intimidated by seeing this chemistry. One of the things I've found in my own experience as a student and now an educator, once you start to apply the fields that you might not be as comfortable with, to what you're doing, it makes a lot more sense. It's not as challenging. By the end of our program, you're really going to be just concentrating in your core labs. You'll be in the lab all the time, probably 4 or 5 days a week. And this is where we're going to start deciding which biotech courses you need to take outside of our required courses and which chemtech courses you might want to take as well. We also have a bunch of different electives ranging from like genetic and molecular biotechnology to forensic science. And of course, there are some in chemtech as well. And the thing that we end with is an internship. And the internship, I find, is the most robust thing that our students do that gives them that bigger picture idea about what they really love to do and whether or not they do want to be in this field or want to maybe look for something else after the fact. But from this internship, our students have found many opportunities, whether it be getting a job when they move on to a university in a lab somewhere, or getting a job either part-time or full-time being hired with their intern, or of course, a company associated with their internship. So it's been really successful. And, you know, it's where we get you guys in the door, you get your foot in the door, you get that hands-on experience, and then you know people, you've networked, and you can move on to whatever you decide. I will say our students have been successful. If they want to continue and get their bachelor degree, master, PhD track, they are doing that. And if they want to get a job right away, we've also found that they're able to do that as well, making very good salaries. So do y'all have any questions about this? Try not to take up too much time. Anyway, if you decide that you want to ask us questions, all of our contact information is here. And of course, it's going to be on some of these handouts we're giving you guys.
Thank you, Ms. Schunick. I am going to take back the sharing of the screen. And I appreciate Deandra and Ms. Schunick for discussing both concentrations of the program. In the chat, I have placed the handouts for today, both the main handout with the presenters, as well as a meet the team, and which that also includes student testimonials, as well as all the teachers. Just a reminder again for those that logged in, please share any information you would like on different social media platforms, Facebook, LinkedIn, Instagram. Again, more contact information that you can use from the handouts, or you can go to our website. And as a reminder, as we move into the presentations today, if you have a question, you can place it into the chat, introduce yourself into the chat, meet the other individuals attending. If you have a question, you can raise your hand, and we ask that you remain mute if you are not speaking. And then there is closed captioning that's available throughout the sessions. So you can click on the CC on the toolbar and the closed captioning will appear. This is a QR code that will go directly to the Science Lab Tech website that Ms. Schunick was showing online. So you can grab that. But again, it is also in the handouts we are providing. So I am excited to show you the SLT open house schedule. Our first guest today is going to be Dr. Nicholas Sandoval. He is from Tulane University, and he has been a great supporter of Delgado Community College's SLT program. He has created an iGEM team that our students are participating in, as well as included us in potential grant funds that will allow students to get some undergraduate research experience in the summers. We are a little early to start for Nicholas, so if anyone has any questions before we move forward, please feel free to ask them. If you have any questions about the curriculum, whether it's chemical technology or biotechnology, we have the two directors on right now, as well as both department chairs. Aaron, you have a question? Go ahead.
Yeah, I'm curious what the internship looks like. as far as, is it one site or multiple sites, and how many clinical hours, and roughly how long does that take full time to complete?
So the internship is equivalent to a full class, which is a 45-hour commitment. Recently, we added a few more requirements that takes about 6 hours of your own time, but it really depends on how technologically savvy you are, kind of things like that. So it's about 50 to 51 hours total. The 45 contact hours means that you'll complete 45 hours of hands-on training with a mentor or, you know, a supervisor of some sort. We have a lot of partners in the city where our students have been working. With the pandemic, that has been a little bit more challenging. So sometimes students work with teachers. You know, I'm having an intern this semester, for example, but typically it kind of works two ways. If students have, like, if you had an interest in something that we really didn't have a connection for, we would look for a job or an internship opportunity, make those connections and see if they wanted to take you on. If we already have something established that you are interested in, then it's kind of like applying for a job. You know, you get your resume together, your cover letter, we reach out to them, or you do, you see if they want to work with you, and it's established that way. And I mean, I'm always willing to make more connections. So if you find someone that you really want to work with, I will contact them on your behalf and just see if we can establish that. But so far, it's been pretty successful, and we've been able to place students at a variety of different places, some of whom are going to be speaking today to y'all. So.
And then Aaron, I just placed just in the screen, it's not full screen, but these are just some of our partners, from four-year institutions to the police force to biotech and chemtech firms, and then chemical industry partners, as well as USDA. We're one of the only schools in the nation to have a partnership with the USDA for a biotech and chemtech program. So lots of good opportunities out there.
How long does it take to complete that 45, 50-hour internship?
You know, it's really up to you. Some students do it in like a month, but a lot of students actually end up completing, like doing more hours, just because they get engaged with the experiment or activity that they're working on. Some students like to do it over the course of the semester, and some will do it very quickly. And we also have a mini session where you can do it in 6 to 8 weeks. But usually it's kind of up to you and your mentor, and if there's a timeline on the project that you're working on. But usually y'all kind of figure that out, and you're going to work with whatever your schedule allows, basically.
Is there a standardized exam after that internship?
No, the other work we added is part of that, so that we can kind of gauge, you know, what skill sets you've acquired, if you have any concerns. And I am the contact person, at least for biotech, for that. So I check in a lot and make sure that you're actually having a good experience and not feeling like you're doing busy work or, you know, something like that.
Cool,
Aaron,
thank you.
in addition, most of our students, and this is true for chemtech and biotech students, they finish their 45 hours, and most of them end up doing 200 to 300 hours because they want to finish the project they are working on. And some of our students get hired on as afterwards with soft money. So when we have grants that are funding a particular project, if the mentors or project manager has money, they will hire the students on for projects. And a lot of our students have stayed on for full-time careers at their internship. And then to give you a little bit more overview of the work that Ms. Schunig was addressing, it's help writing your cover letter, your resume, networking, Excel skills, interview skills. So everything to make sure that you're a well-rounded individual with both technical skills and human interaction soft skills.
Thank you.
You're welcome. So it is 9.
Well, okay, we're ready to go. Never mind.
It's 9:29, and I'm excited to introduce our first speaker. I briefly introduced him earlier. This is Dr. Nicholas Sandoval from Tulane University. I am going to stop sharing my screen and give him hosting privileges. So big round of applause for Dr. Sandoval. Oh, and he just got kicked out, so I'm letting him back in. It's COVID and it's technology. We all know how that works.
All right, can everyone hear me and see my screen?
You're good to go.
All right, well, great. So I'm doing this from my car. The boy is currently at the pediatrician's office, so I'm trying to, hotspot it and do all this type of stuff. So, yeah, we'll roll with it. So, thank you for the introduction. My name is Nick Sandoval. I'm an assistant professor in chemical and biomolecular engineering at Tulane. And in the lower right-hand corner of the screen, you'll see a very outdated picture of my lab. And if you look over my right shoulder, you might see a familiar face. That is Dr. Claire McGraw, who's actually now faculty at Delgado. So she was a postdoc in the lab, and now is teaching at Delgado. So, and we miss her very much. Just chatting on the Zoom chat. So very briefly, the Chemical and Biomolecular Engineering Department at Tulane has these faculty members. We do a lot of research in a lot of different areas, not just biotech. So I'm obviously in the biotech area, bioprocess and biomedical. We have people who are heavily involved in materials, energy, and computational simulations. And many of those people overlap in multiple bubbles. So just a brief overview of how I got to New Orleans, and in particular, how I got to Tulane in the chemical engineering department was, so I grew up in Fruita, Colorado, small town on the border of Utah. I went to both undergraduate and PhD at the University of Colorado Boulder. After that, I taught for a year at a public university, Colorado Mesa University, out near my hometown in Grand Junction. After a year, I actually got hired as a postdoc at the University of Delaware, all the way across the country. And then after that, I spent 4 years there. I got my current job here in New Orleans at Tulane. So what do I do as a research-oriented professor? Well, I'm a professor, but actually not most of my job is teaching. It turns out that I would say around 60% of my work is research. That is, we write grants to fund our projects. We think of ideas for those projects to work on. And then if we're lucky enough for them to get funded, we hire and train graduate students and postdocs. I oversee the day-to-day lab operations, ordering chemicals, ordering materials, advising graduate students, having meetings, analyzing data, talking about different experiments we might want to do. And then after we're all done, hopefully that leads to some pretty cool journal articles. That is, scientific papers that are sent out to editors to be published in scientific journals. And then on the side, when you're in the scientific community, it's all about that community. So what we do is we review other researchers' grant proposals and journal articles to make sure their science is sound and their ideas are really cool and worthy of federal funding. And then normally in non-COVID times, we have lots of conferences to go to where we talk about our work. So I've been able to go to amazing places like Cincinnati, Ohio, and less amazing places like Munich, Germany. So it ranges all in between there. 30% of my job, I would say, is about teaching. So we actually teach courses that involves, doing the lecture, grading homework and exams, hosting office hours. And then for the larger classes at Tulane, I get teaching assistants who are graduate students. And then, so I oversee those, and we prepare new courses. So one of my fun things that I've been able to do in this time is not just teach the courses that are standard in the program, but develop my own about genome engineering. And then the last little bit of my job is about service. So I advise student organizations such as iGEM that was mentioned before, which biotech students at Delgado are more than welcome to join and do actual wet lab work. And then I serve on various university committees, which is a lot less. So the path to being a research-oriented professor is pretty long. So what you do is you get an undergraduate degree. For me, it was in chemical engineering, a Bachelor of Science, that took 4 years. And then after that, or during your last year, you apply to PhD programs around 6 months from the finishing. And so then the PhD program is in chemical engineering for me, but if you decide to move on, it could be in a myriad of things like chemistry, microbiology, or other programs. That takes around 4 to 6 years. And during the last year now, it's becoming more and more common that people need to do a postdoctoral position, just like Claire did in the lab. So the postdoc generally happens from 2 to 4 years. That can range. There's no degree at the end of a postdoc. It just stands for postdoctoral research. And when you get to sort of the end of finishing your postdoctoral research, you can apply for faculty positions. And this is what's interesting. You apply to very many schools. I applied to like 59. You get interviewed at a few, less than 10. So I got 5 interviews. And then you get offers, if you're lucky, at even fewer. And then you hopefully get to choose between 1 or 2 schools, and you pick the one that you like the most. So I think now I'm at a pretty good stopping point. I see there's one hand raised. So I'm not sure if I have to allow anything, but go ahead with your question.
Dr. Sandoval, that's really a very nice presentation. You asked the question. I'm interested in a microbiology research project with our students at Delgado.
Okay.
And I have been trained from Tulane Medical School as a postdoc. So is there any opportunity for our students? They can initiate some microbiology project with you?
Yes. And the very quick and easy way to do that is the iGEM. So iGEM stands for International Genetically Engineered Machine Competition. It's universities around the world take part in iGEM. And just this past year, I wanted to start a synthetic biology team at Tulane. And in that process, I thought there's no reason it should be limited to Tulane students. So I contacted Dr. Rosenzweig and said, can we have a partnership? And so now Delgado students are more than welcome to join iGEM. And so with a team-based project, we work in the lab. We do wet lab experiments and try to solve a small research project. This year, we're trying to optimize the short RNA sequence called a CRISPR RNA to work with a protein called Cas12a for improved genetic editing. So we're making mutations in the lab. We are doing microbiology. We're doing a lot of cloning. And hopefully by the end of the semester, we'll actually be doing experiments.
Yeah, I mean that it will be a very small project, so they can at least they can present at the medical school level or the university level or the college level for the seminar and the workshop. They can present as posters. That will be really big tool for them.
Yeah, it's a large part of the iGEM competition actually is how you present the work. So a large part of it is scientific communication. So I think it's a perfect.
So I think that our biotechnology and with Dr. Rosenzweig that we can get in touch with you to support our students.
Yeah, yes, please. And I unfortunately didn't put my email up here, but
I have it with me.
Oh, perfect. Well, then any student can just email and we can get them involved in iGEM pretty rapidly.
Thank you very much.
Let's see. So I'm coming up on my 10 minutes. Let me just talk about maybe one type of project that we do that's common in our lab. And I'll go to Dr. McGraw's project because she's in the Zoom room as well. So one of our projects is a collaboration where we try to study the effect of bioprocess stresses in the production of monoclonal antibodies. So monoclonal antibodies are the proteins that are used as biopharmaceuticals. The most common commercial I see on TV is for Humira, and that's a monoclonal antibody. So you can see kind of like a cartoonish image of a monoclonal antibody on the right. And one interesting thing is that these MABs, that they're called, monoclonal antibody MAB, need to have sugars placed on them. And this is why they're kind of difficult to make. And they're produced in specialized cells called CHO cells, Chinese hamster ovary cells, because CHO cells can make these sugar structures a lot like human version of those sugar structures, so they don't cause an adverse immune response. So one of our research projects was to figure out how much of stress can these cells endure and still have good sugar glycosylation patterns. And so you can see her work right here shows that when these cells are undergoing ammonia stress, which is a common bioprocess stress, you get less glycan maturity, meaning the structure of these sugar compounds is less complex and thus not as high quality of a biopharmaceutical. And so a related project is what Dasha is working on, where she tries to engineer the CHO cell itself, the genome, to have improved genome stability and product quality by editing the genes using the technique called CRISPR, which you may have heard about. So I think that's my time to talk, and I think the rest of it should be for Q&A for anyone who has any questions.
Nick, I don't know if this is not considered okay to ask, but do you mind saying a sentence or two about the potential grant that you applied for, that Delgado is a partner? Is that okay to talk about?
Oh, yeah, sure. So the way we actually pay our graduate students, because our graduate students get paid, that's another thing that's not necessarily widely known. When you go get a PhD in STEM, you don't have to pay, they pay you. They don't pay you a lot, but they pay you. So PhD students and postdocs get paid. The way we do that is we write federal grants and we ask the federal government for funding to tackle a project that's of national need. So our project that we just wrote a grant with Delgado and the University of Delaware was to engineer bacterial cultures, bacterial strains, so that no carbon dioxide is released when they eat sugar. Normally, carbon dioxide is released when glucose is eaten, and you lose 1/3 of your entire carbon to carbon dioxide, which is not great efficiency. So what we were aiming to do was involve multiple bacterial cultures, multiple bacterial strains. One of the strain actually consumes carbon dioxide as its food source. So we proposed a project to where we can get 0 carbon dioxide release when these cells are consuming the sugar, so that if we have waste biomass and waste carbon dioxide, what we can do is convert all of that to biofuels or other valuable chemicals. Notably, I think in this proposal, it was isopropanol, which is actually quite expensive. And so we're aiming to convert these waste products to valuable chemicals through biological processes. And so that, again, involves a whole lot of genetic engineering, a whole lot of systems biology, and a whole lot of lab work. And then the part that involves Delgado was getting Delgado students to come to Tulane to work over the course of the academic year and the summer to help in this research project, as well as aid in development of biotechnology modules.
Could that be used on a larger scale in the future to reduce carbon dioxide and carbon, emissions and such, like on a much larger scale?
Yeah, a large part of that grant was the thought process that you could hook these type of reactor setups, these biological reactor setups, on the back end of chemical plants or other type of plants that have large carbon dioxide emissions. And so if you were to do that, you could run the effluent gas from whatever industrial process it is through these bioreactors in order to get carbon consumption. So yeah, this does have the potential for large-scale carbon sequestration.
Dad, you changed.
Well, Nick, thank you so much. We appreciate you coming and presenting to us. And thank you for making it work, especially with you having to take your son to the doctor. If y'all have any questions for Dr. Sandoval, you can reach out to me, Ms. Shunick, and we can give you his email address. And we do have a great partnership with Tulane. So again, wonderful opportunities for students that are participating in the SLT program. Again, thank you so much, Dr. Sandoval. We appreciate you coming.
Thank you.
It
And
was a
our
pleasure to
next
talk to all y'all.
presenter.
Yeah.
Thanks, Nick. Our next presenter, let me get my screen. Ms. Shunick, if I don't pass them, Ms. Shunick is going to introduce our next presenter.
So next up is Dr. Wendy Schluchter, who is the chair of the biology department at UNO, and also a big supporter of the SLT program, and someone that has offered us several internships. So I'll let Dr. Schluchter take it over. Thank you so much for presenting today.
Sure, thanks for having me. Can you hear me?
Yes, ma'am.
Okay, great. I'm going to apologize in advance if my dog barks. These working from home days are always interesting, aren't they?
I think if your dog makes an appearance, you get bonus points.
Okay, well, we'll maybe try to do that because he just had a bath, so he actually looks cute right now. So, hi everyone. I'm Wendy Schluchter. I'm the department chair in biology at UNO. And I'm just starting to talk a little bit today. I have slides, but I just decided I don't want to bore you with a bunch of hardcore data. Not that data's boring, I find it very interesting, but I wanted to really talk about, from your perspective, why you might want to go into doing research in a lab. And I would say let me start by saying that when I was in college, you know, I wasn't sure what I wanted to do. I really liked science, but I really hadn't had the experience of actually working in a lab other than the lab classes I took. And it was really a key thing in my development was taking a summer and going to Case Western Reserve University in Cleveland, Ohio, to do a research project with a faculty member. And that's kind of when the light bulb went off for me that I could have a career doing research and making contributions to science from a very different perspective than, for example, going into medicine or going into veterinary science or something like that. So for me, it was kind of a really pivotal moment. And so I think as an undergraduate, I would encourage all of you to try to get opportunities outside of your classwork to do research. And you just heard about some great opportunities from the previous speaker, which is fantastic. And there's lots of them out there. At UNO, we actually highly encourage our students to all get research opportunities, even if they don't think they want to go into research. And we do this in a number of ways. So we allow every student in their biology degree to count up to 6 credits of independent research towards their biology, towards their total 40 credits that are required in biology. And so that allows them to do research with a faculty member over at least a year and get credit for it. And I think honestly, of all the students that I've had go through my lab, all of them have said that research experience was probably one of the most important things for them to connect what they learn in the classroom to what they could actually do with that information. Sometimes you think as professors, we're telling you all this stuff, and you're like, why do I have to learn about photosynthesis? Well, there's lots of good reasons, but what you see is, you see it put into action in the lab or in the field if you do field research. Excuse me. So that experience as an undergraduate kind of opened up the door for me to think about graduate school. And you just heard a little bit about graduate school from the speaker from Tulane. And the nice thing is that if you decide you're going to get your bachelor's of science in science, you get hands-on experience, then when you apply to graduate school, you're going to be kind of one of the students that has that experience that they want to accept into their program. And so at UNO, we have a bachelor's in biology, we have a master's in biology, and we have a PhD in integrative biology. And so I have graduate students at all levels in my lab, and I have undergraduates. Right now I employ, I think we have 4 undergraduates in my lab. 2 are earning hourly wages to do that. They're getting paid $15 an hour to work in my lab. And then 2 are doing it for research credit hours for their degree. And so the undergraduates work with the graduate students, and they mentor them to really figure out how, learn the techniques, make sure you have good procedure, so that, you can repeat your results and have confidence in any results that you get. The other thing that's really critical, I think, is that you learn how to present what you've done to an audience. And so that's really an important thing in most of these research experiences that we give undergraduates, is that we have you every couple of weeks or a month or whatever, there's usually a lab meeting where everybody in my lab gets together and we take turns presenting. So undergraduates, graduate students, and sometimes I present on the latest that they've done. And it's more about getting used to talking in front of a group, because it's very nerve-wracking, especially for students. But the most important thing, I think, why students should have to present their work, either as a poster or as a little talk, is because you learn how to explain what you've done. And honestly, that's when you really get what you've done, is when you have to talk about it to other people. And so that's a really important component of research and lab science, is to really discuss your results, kind of share them with others, and get used to that, get over those little nerves that we all get, and share what you've done with the larger community. All of the students in my lab present their work, either certainly at lab meeting, but also at conferences. And so we have several conferences that are here locally that you can go to, and then the best students get invited to go to national meetings. And so, for example, I took 2 undergraduates with me to a conference. It was actually an international conference, but it was held in Chicago, and they were the only 2 undergraduates there. But I was so proud of them. They did such a great job. And both of those students are now in PhD programs. One's at Stanford and one's at the University of Washington, Seattle. So these students are as good as anyone else, anywhere, and they got their degree at UNO, and I'm very, very proud of them. So what I want you guys to think about is that research is an option for you. It can also be a career. For example, you could get your bachelor's degree and get enough experience while you're an undergraduate to go get a research technician job at downtown at LSU, or in one of the companies that is here locally that hire our undergraduates. Eurofins is one example of that. We have several of our undergraduates that are currently working at Eurofins. It's right at the lakefront, and they do molecular analysis of food that passes through the port of New Orleans to verify whether they have genetically modified grains or not, and whether there's certain chemicals in the food that are harmful. And so they are really important in assuring food security and being an outside sort of verification of that food security for companies that are passing their products through the port of New Orleans, for example. So there's lots of opportunities to get jobs. If you don't want to go to graduate school, that's fine. As long as you get sufficient experience as an undergraduate, you can really translate that into a job. But the problem is a lot of undergraduates are so busy with their classes, they don't think about those opportunities, and they're really out there. You heard about this opportunity to participate in this iGEM program, for example, that's a great opportunity. UNO has summer opportunities for undergraduates through the AMRI program, which is Applied Materials Research Institute at UNO. They have an application for undergraduates from all over the country to come to UNO in the summer, but they also potentially will consider community college students to get supported for the summer to do research with the UNO faculty member, and I'm a part of that program, for example. So just to tell you a little bit more about myself, I went to a small school for undergraduate, did this summer research program, and then realized that I could go to graduate school and they would pay for me to go to graduate school, which was a revelation because my family certainly didn't have enough money to send me to graduate school. And so I was able to support myself to get my own, to get my PhD at Penn State University. And then I did a postdoctoral fellowship, just like the gentleman from Tulane told you about, at UC Berkeley. And then I got a job at UNO and I've been here ever since. And I think that the thing that I really enjoy about being at UNO is that I'm able to teach classes, but also have a research lab. So it's like the best of both worlds for me, where I really get to interact with students. Teaching keeps me thinking about the big picture and not just my little area of science, but research also keeps me reading the literature so that I keep learning as I go. And so once I got this job at UNO, I realized that there was just so many undergraduates that would benefit from getting that hands-on experience. And so that's really been an important overarching theme throughout my career, is to involve as many undergraduates as I can. And so not only do I involve them in my research lab, but I have also applied for grants to bring undergraduates and involve them in many different labs throughout UNO. And also we had a partnership with SUNO as well. So we've, and then, you know, I've definitely mentored undergraduates from other universities, including Delgado. So there's always opportunities. If you think you're interested in that, one thing that I would always tell students is, you can always go on a website of a university and kind of look at faculty members' web pages to see what they do. And if it's something you're interested in, you know, kind of do your research, learn a little bit about that area, and then write that person an email. I get emails from students and I always try to respond. I've mentored high school students that have reached out to me. So it's never a bad thing to always try to communicate and try asking about opportunities to get involved. And so I would encourage you to do that and stay focused on learning the skills, the best skills that you can do in this program, where you really understand what you're doing and why. That's probably one of the most important things. And the other important thing that I tell my students is, don't be afraid to make mistakes. You're going to make mistakes. The only thing you can do is learn from them and try not to make a different mistake tomorrow. And if you do that, you're always going to be moving forward and making contributions. So I'm going to stop talking now and see if there's any questions at all. I can talk more about my research, but I thought I would give you more of a general view about kind of how UNO can help get you guys involved in research.
Are there any questions?
Dr. Wendy, can I go through?
Sure.
This is Dr. Ansari from Delgado.
Mm-hmm.
And I have been listening before to you and the presentation. And our student from the microbiology, they are interested in doing a lot of the research work.
Mm-hmm.
And maybe in the coming semester, we are expecting a few students, they are going to opt the research work. So basically we wanted to expose our student for the larger community to go through the lab exposure or to the kind of the local conferences. So this is our main motive too. So we need your help in this connection. I really appreciate if you can give some opportunities to visit your lab
Sure, we
and
can do it
discuss the projects.
Yeah, we can definitely talk about that. We just have to, we're in strange times right now. And so we just have to make sure that we have only a few at a time and we figure out how to spread people out. That's been the challenge. But we've been back in our lab since the summer, but we've basically put people, my students are sort of trying to work different shifts and spread people out just to keep everyone safe. So that's the only caveat there, is that we'd have to figure that out. But absolutely, I think the best way you can excite students about what they're learning in the classroom is to get that hands-on experience, and they can see how that information is important beyond just the tests that they're taking.
Yeah. Now, one thing is that we wanted to make our students familiar with the research instruments, research equipments, what is their functions, how it works, what is the mechanism, all these things as will be. So just the visiting to your lab also, I think that is going to be a big tool for them.
Right. Yeah. And I think just not being intimidated by an instrument that you don't know a lot about, because all of us can learn how to use it, and just being willing to do that, I think, is really important.
Right. Thank you very much, and I will certainly be in touch with you.
Okay.
Thanks.
Thank you so much for presenting for us today. I just wanted to say a quick word. I put this in the chat, but we are working directly with Wendy and UNO and another department on a grant that we should hear about pretty soon. And this grant focuses on a new concentration in a major at UNO, but the grant also has Delgado as a 2+2 pathway, specifically focusing on the SLT program. So lots of opportunities are out there right now with research. And again, UNO, Dr. Wendy has been so supportive of the SLT program and its growth. So thank you so much for presenting to us today.
Sure.
So next presenter is Dr. Ike Levine, and he is from Maine. And we were very fortunate enough to meet him right before COVID. He came to Delgado and did a huge presentation on his field, which is algae. And we all fell in love. And so now we participate with this program. We're partners, and I'll let him explain all of the opportunities available. Thank you so much. Ike, do you need host privileges to share? Are you just going to talk?
No, I have the PowerPoint that I sent Charlene yesterday, or if you give me screen sharing privileges, I'll pop it up myself.
You are now co-host.
Is that a good thing?
Aha, yes. Take over. Make us smile.
This will stop other screen sharing. Okay.
Yep, which is fine. Let me stop it real quickly.
All right. There we go. Can you all see that?
Yes, sir.
Oh, excellent. Well, thanks just an awful lot for the opportunity to chat with your students. And I couldn't agree with the previous speaker. Wendy was dead on in terms of just build a relationship with a professor one way or another, and it will be the basis for the rest of your life. And for me, it was Janet Benamo at Cornell. And I just went up to her and said, can I be a teaching assistant? And I wasn't the best student in the world. And she took me in. And that was 1975. And I've been involved with teaching ever since. But today I'm going to chat about the Algae Foundation. I'm both a professor at the University of Southern Maine, but I'm also the president of the Algae Foundation. And we've raised, I don't know, $6-7 million to develop, develop the training programs necessary to fill bioeconomy jobs. And so we're gonna talk about that. Let's see, how do I advance this? Oh my goodness. Oh, there we go. And so we get our money from the Department of Energy. And the Algae Foundation, along with one of the National Renewable Energy Lab, formed ATEC, which is the Algae Technology Educational Consortium. And Delgado is now a signatory and an official member. And what we've done is we've developed a series of interest generators. They're in green, and then formal curricular programs, which are in white. And so we'll go through most of them. Our program or our partnership with Delgado has to deal with biotechnology. And so as you can see in the white boxes, we have degrees and curricula for algae biotechnology. We have one out west for algae farming. And then we have more informal learning, an extension course, or a series of extension courses called ACES. And in the end, we've also just developed a badging program, which allows students to test out and prove that they have specific skills. And once they pass these exams, they are put on a national register of skills. And so when you go to a job, instead of just a resume, and we all know people can write tons of stuff on resumes, but you have these badges, which a potential employer can go on the national registry called Acclaim, and they can look you up, and it could say, I know microscope skills, or I know extraction skills, and it will say exactly what you've been certified to do. And that takes a lot of uncertainty out of looking for a job. Okay, and just a quick review. We have Delgado, as you see where you are, but the red and the green schools represent our formal group. This is a slide that was prepared for the DOE, and we get reviewed every 2 years. So the green circles represent our older members, and the red circles represent our schools that have signed up with us since 2019. And we've signed up our first international school out of Korea. And you'll notice that these schools are almost exclusively on the coast. And that, fortunately or unfortunately, represents where the embracing of algae happens to be. And our first effort is the Algae Academy. This is our K-12 effort. And we provide, and everything that's on this presentation, everything that the Algae Academy does is 100% free to all students, all teachers, all schools. And so we started about 4 years ago. You'll notice, I don't know, can you see my pointer? Well, whatever, even if you can't, you could see the tremendous growth. This year, 2020, 2021 is only a partial year so far. So we're still, even with COVID, scheduled to do very, very well in terms of the number of students. We've reached about 75,000 K through 12 students around the country. And we pivoted starting last summer to develop this in-class effort to be an online. So about 50% of this year's academic participants are through online programs. And we also developed these Summer Science Institute called SASE. I guess that's the old name. We had to change it because someone pointed out that it's SASE. I guess that doesn't go over well. So actually, it's now Summer Algae Science Institutes. And we bring students, and last summer we had one scheduled at Delgado. Hopefully it will be offered this year, where we bring in teachers from K through 12 in the neighborhoods and who already have relationships with our partnering community colleges. And we teach, we train the teachers to be more knowledgeable about algae and how to offer the curriculum. So hopefully, if it's this summer, either in person or online, people who are watching this can be involved. The second interest generator is what we call MOOCs, Massive Open Online Courses. It's a platform about 15 years old, started by Harvard and Stanford and Princeton, where college professors put on courses and they're offered free of charge around the country. And so this is the graph you see, and you'll see the big spike exactly 1 year ago, represents the weekly total of new students. And we were averaging between 50 and 100. And once COVID started, we reached the maximum of 450 new students in a week. And so we've reached over 17,000 students. But the relevant thing to everybody listening here today is 10% of those 17,000 students received either a promotion or a pay increase as a result of taking our course. So that's from a relevance point of view, that's very real. Our second one we've just published just about a month ago is Algae Biotechnology. And the photos on the bottom represent some of the subject matters. As you can see, we have, in the intro of algae, we talk about farming and biology. But on the right-hand side, I bet you didn't know that now surfboards and flip-flops are made out of algae. And so this is a picture of one of the surfboards made out of California. The colored stuff, the purple or the blue in the middle, is where all M&Ms come from now. All M&Ms will be naturally colored by pigments that come from algae. And so algae has a tremendous amount of utilization, and you might want to consider it as a future career path. And so the next series of slides is how what we do affects you guys in terms of potentialities at Delgado. We have created a series of lectures and labs that can be incorporated into your biotechnology classes. And we give your professors the laboratories. And then, but some of them are tremendously complicated. And so recently, we just started to complete what we call our IGSOPs. And the IGSOPs, image-guided standard operating procedures, go over in meticulous detail how to run the laboratories. And so even though we may not be at every school to personally offer the curricula, that with the IGSOPs, both your instructors and the students can have a much greater understanding of the whys, the hows, and the wheres in terms of performing these laboratories and developing skills. And our skill sets, what we try to teach, comes from our industrial advisory board, which means it's industry who is saying, well, we really need these skills in our new students. And so when our students graduate, they have the skill sets all ready to be productive employees or entrepreneurs. And so industry has really responded well to our efforts. These are some of the names of the different courses that we insert our individual laboratories. Many of you may have taken some of these already. This is a somewhat old slide because the number of students who have taken it is now up to around 1,500. And this is just what one of the labs looks like. This is reasonably close to you guys in Austin, Texas. This is the Austin Community College, and they have a robust biotechnology program. And this is barcoding. This is using algae that they grew up and then extracting genetic material and going through a series of analyses. And so, again, imparting great skills that makes them much better appropriate for applying for jobs. Additionally, we have created new courses. These are the two levels, represent two separate 1 credit intensive labs. They start at 7 o'clock on a Monday morning and they go to around noon on Saturday, and you earn a single college credit. But each one of these efforts that are involved here are singular skills that are necessary to be biotechnology lab professionals, even if it's not based in algae. So when we were reviewed by the Austin Community College External Advisory Board, they realized that learning about algae makes the students better prepared to be in any industrial, whether it's mammalian, cell lines or bacteria or starch. Having the more fundamental knowledge gleaned from algal-based labs is very, very helpful. We also provide free textbooks. And so this is a PDF, a downloadable PDF for all ATEC students, free of charge. In fact, we got the material from my publisher from 2 books I produced, and they gave us a fee-free license to extract the biology of microalgae and the biology of seaweeds and put it in one publication and give it away free. So kudos to Academic Press for their generosity. And we talked about the badging and why that's really important. It's amazing how well, how quickly this has been adopted. This looks like a complicated slide, and it just lists 4 of the many badges that we offer in different skill sets. But what it provides to the student is a way to market themselves, but also provides them with a tremendous digital platform of where the jobs are located, what are the skills needed by each job, how much they're paying, and what are the companies. And so that is an amazing asset. On the other hand, when you walk into an employer, you give them your badges. They can then look at this page and say, for this singular badge, the list in blue are the skills needed. And so when you hire this person, they can, the employer knows, well, they certainly know different kinds of algae. They know how to work in a lab. They know how to work in fundamentals of clean technique, lab safety. And so it takes the guesswork out of hiring. So this is tremendously valuable. And from the student point of view, you look up, you look at one of the keywords in this example here. The keyword was algae. It shows you where the locations for jobs are, what are the salaries, what are the names of the companies that are hiring, and the number of jobs available around the country. So this is turning into a tremendous priority for the Algae Foundation to build more badges, both in farming and in biotechnology. And again, another partner of ours, Lone Star, north of Houston, is developing a biomanufacturing course. And built into that biomanufacturing course are these 11 micro badges. And again, you show up at a potential an employer and you hand them your 11 badges. This tells them you are really skilled. And it's not because you said you're skilled, but you have been certified by a nationally endorsed organization to offer these badges. So again, I was never a Boy Scout, or a Girl Scout, but if you remember, they get those sashes with all the badges, and they're all tested out and proven. This is exactly the same concept. And how else is algae going to help you in terms of a career pathway? We have partnered with the Department of Energy. They have a bioenergy career exploration wheel, and on it, embedded in it, is all of our efforts. And again, it shows you on the top here different levels of curriculum that we offer, and the DOE embeds it in their website in terms of helping people learn about jobs, learn about career pathways. And if you're ever interested in following more about biology or farming or biotech, this is a tremendous place to start. And so I'll open the rest of it up to questions, but I couldn't have agreed with Wendy more. My lab, I sold a company and retired for a while, and then I came to the university, and they had an empty shell of a laboratory. I brought my equipment, and my agreement with the university is I could do private research, I could do basic research, but as long as I train undergraduate and graduate students. And I have people come from all over the world to train in the lab. And it's amazing what a kickstart that is to either an academic career or a commercial career. And there are great jobs in both. So I open up the floor to questions.
Dr. Levine, we did have a student state that she already started one of the MOOCs. So
Oh, great.
yeah, so this is exciting. And we got our algae in at the beginning of the semester. So we are growing our algae now.
Super. That's great.
Does anyone have any questions?
And, nobody grows up thinking they're going to be an algae expert. I certainly didn't. You want to be a doctor, an astronaut, a firefighter, whatever. In fact, I was in medical school and left. Well, I left to play rugby, but ended up in graduate school and discovered algae. But about 5 years ago, I went from being a science nerd to being cool. So now I'm cool because algae's cool. But algae, it can represent a tremendous life. It's taken me around the world. I have colleagues everywhere. Let's just say it's not something you inherently think of, but again, when you take a deep breath, just remember 50% of all the oxygen you just breathed in came from algae. Think about all the oil that you've ever pumped in or gasoline you've ever used comes from dead algae. And so think of any chalkboard you've ever been in your class. That's dead algae. And so in terms of what algae can do for the human condition in terms of biodegradable plastics, biofuel, blue M&Ms, and it's a remarkable endeavor, whether you think about it or not. Or it certainly is something to consider if you like exciting new pathways for your future.
Well, thank you so much for sharing your knowledge, attending, and allowing Delgado to participate with you and helping our students grow and learn. We appreciate it. If anyone has any questions for Dr. Levine and you didn't get to ask them, you can contact me and I will provide you with his information. So next up, I'm happy to introduce not only a good friend of mine, but someone that has been a huge supporter of Delgado and has helped us form a partnership with the USDA, and our MOU with the USDA is almost complete. I would love to introduce Dr. Jackie Nesbit. And Dr. Nesbit, I forgot to ask you, do you want to share the screen or are you just talking?
I'm going to share the screen. I have a little PowerPoint.
Let me give you privileges. Okay, you should be able to share. I'm going to stop sharing, Jackie.
Okay, got it. Yeah. I'm going to pull up my PowerPoint.
And while Jackie's pulling that up, just to let y'all know, Dr. Nesbit is also a faculty member at Delgado and UNO.
Well, thank you. I kind of hate to follow Ike now. Next time, can you schedule me if we do this again before him? It's kind of a hard.
I will keep that in mind just for you.
Can I pick my time slot next time?
Yes, ma'am.
Thank you for inviting me to speak. My intent for this talk is just to kind of walk you through my education path, along with some of the techniques that I've learned in the variety of labs that I've been through, kind of because I did all of my education locally. And so I think it'll give the students kind of an idea of what the opportunities are in terms of learning different skill sets in different laboratories around New Orleans. My education, or my path to where I am at the USDA, is kind of a non-traditional one. I didn't get my PhD until I was in my mid-40s. So I had always loved biology from the time I was a little kid. I was fascinated by the processes of life, all of that. But when I turned 18, I kind of thought, oh, what am I going to do with a biology degree? And so it didn't really seem practical. And so I did accounting. And when my kids were starting school, I was a stay-at-home mom, and I thought, oh, here's my opportunity to go back to school and get my biology degree that I always wanted. And so that led me to the University of New Orleans. And one of my first experiences with research there was through Howard Hughes Medical Institute, a grant that they were offering at the time to undergraduate students. It was $2,000, and you basically could sort of write a mini grant where you picked a laboratory and you came up with the question and you just designed an experiment, and then you wrote up your results. And so I participated in that program, and that landed me at the USDA here in New Orleans in the lab of Dr. Raja Sakaran, who was studying the effects of this very potent toxin from a fungus that infects lots of our crops. And that's called aflatoxin. And so I learned a lot of different techniques in his lab, and it was kind of exciting because I got to use this gene gun, which plants are very hard to transform, put foreign DNA into them. And so you have to shoot it into the plants. And so this is called a gene gun. And so Dr. Raja has this gene gun in his lab, and I got to use it. And I also learned how to culture plants. So it's very difficult to grow a plant in a petri dish. And so I learned that technique, and also got to play around with liquid nitrogen. We had, ground up these tobacco plants. So we were studying it in tobacco. So that was a really interesting experience early on in my career. And I took the $2,000 and bought a computer. And another experience that I had at UNO was with Dr. Mary Clancy. This is directly from the UNO website. She's a very private person, so it doesn't surprise me that she does not have a picture in her online. But Dr. Clancy works with yeast, and I worked with her for about a year, the last year of my undergraduate. And so I got to learn how to not only pour plates, make plates, but grow yeast on petri dishes and in flasks. And she really taught me how to clone. So I got to play around and manipulate with a lot of DNA sequences, and that was very fun for me. And she was also very influential to me because when I graduated, I was still working in her lab, helping her out. And she said to me, well, what are you going to do now? And I said, I don't really know. And she said, well, why don't you go to Tulane, to the Department of Biochemistry? They're looking for graduate students. And I had a mantra at the time, never say no out of fear. And of course, that scared the bejesus out of me. So that was a yes. So I ended up at Tulane University School of Medicine in the Department of Biochemistry under my mentor, who was a pediatric hematologist oncologist, Dr. Hemingway. And with him, one of the first things I did early on in that lab, surprisingly, it worked very easily. And it was a yeast 2 hybrid, where using this system, and I don't really have to explain it, but using the system, I was able to identify this interaction between these 2 different proteins. And so that was a really cool technique to learn. And it provided some very valuable information. And it was really exciting to have something work right away in the lab, which the second part of my dissertation or my PhD work was to try to purify that protein AF9. And that took the next 6 years. So that didn't work as well. So it's important to, stay with your research. A lot of things, sometimes things will work and sometimes they don't. And you just have to be diligent. I also learned there how to culture mammalian cells in flasks and petri dishes. So that was very a nice skill to learn. After I graduated, I started working for Dr. Sohaila Maliki, and I'm still working with her. We are at the Department of Agriculture on Robert E. Lee Boulevard in Lakeview. And we study peanut and tree nut allergies. And so a lot of the techniques that we use in the lab involve protein. And so we do circular dichroism spectroscopy, which is a way to look at the different secondary structures of a protein. And we also do some fluorescence activated cell sorting, or FACS, where we're basically selecting specific cells out of a population of cells by just labeling them and shooting a light through them, and we're able to just sort them. So we do a lot of that. And because we work in allergy, of course, we work with antibodies. So we do a lot of different techniques like Western blot and ELISAs. One of the cool things that we're working on now, and I'm heavily involved in, is using these peptide microarrays. So basically what we do is we put parts of all of the different allergens. All allergens are proteins. And so we put these pieces or parts of proteins on these glass slides. So each of these long rectangles just represents these long glass slides. And there's, thousands of pieces of these proteins on these slides. And we take the sera of patients that are allergic to peanut and tree nuts. And the sera is just the liquid portion of the blood where their antibodies are. And we incubate them with these slides. And then we label the antibodies, or we do it with secondary labeling. And so we can basically see the two different types of antibodies that are involved in allergic reactions. And that is IgE on the left, the green, and IgG4, which is on the right in the red. This is just a zoomed in picture of it. But basically we can look at the differences in peanut and tree nut allergic individuals binding, what they're recognizing specifically in the protein. And so of course we can throw a lot of questions at this data, like are there differences between regions like the United States and Israel, or the United States and Australia? We have sera from individuals from all over the world. And we can also, and what I'm looking at is to determine cross-reactivity. So why is it that a person that is allergic to peanut is also allergic to tree nuts? Is it some specific part of a protein that both of them are recognizing? And so it's, and that's just a few of the questions. I could go on and on about that, but that's pretty much what I'm looking at, is trying to determine cross-reactivity between peanuts and tree nuts. Another cool thing that we are involved in our lab is, I don't know if anyone has seen, I think it was last year that this came out, where the FDA approved a treatment for peanut allergy. It's called Palforzia. And this started with just two guys in California that thought, hey, I wonder if we give peanut allergic individuals a little bit of peanut at a time, and then increase the dose if their peanut allergy would go away. And that's essentially what happened. It's now, a multi-million-dollar company. But we did all of the wet lab technique because it was just a couple of guys that started this company, and they didn't even have a laboratory. And so to get a drug approved through the FDA requires a lot of stringent experiments. And so we made sure that every dose, even though the pill is just ground up peanut powder, we made sure that every dose had the exact same amount of not only total extract of peanuts, but also the individual proteins that are in that extract. And so we were heavily involved in that. One thing that I want to mention in terms of, I really like this example of what Dr. Gideon Lax did at King's College, because it's a really good example of just the scientific method in action. And it's the reason that the two guys that started Palvorzia thought about giving people increasing doses of peanuts to cure a peanut allergy. And what Dr. Lax did at King's College was noticed. So he observed the differences in allergy in different regions. And specifically, Israel has a very, very, very low prevalence of peanut allergy. And he wondered why. And when he looked closer, he noticed that the parents of peanut allergic or parents of children in Israel feed their children these snacks called bamba. I think you can get them at Trader Joe's now. But basically it's a Cheeto, but it's made of peanut. And so to kind of relax their children, they keep it in their hand and they just munch on it. They're babies. And what the question that he asked was, wow, maybe it's this early exposure of peanut that prevents peanut allergy. And so he started this study called LEAP, and this study has changed the recommendations for early introduction to peanut all over the world. So now pediatricians recommend that children are exposed to peanut by 4 months of age and no later than 6 months, because it decreases their chance for developing a peanut allergy. So that's just an interesting use of the scientific method, where you just notice something and you can change, make big changes. I'll just end on this slide. This is Dr. Edwards at the USDA. He came up with a gauze that has the ability to increase the clotting process of a wound. And so these gauze pads are now used by all the different branches of the military, because they decrease the amount of blood loss. So he's been highly recognized for that contribution. On the left there, I'm just showing all the different research groups that we have at the USDA, because we are involved with the SLT program. And I think we've had about 6 or 7 students, Amanda, come through. And I think half of them had ended up to be a paying job. So they stayed on after and, and started making money. And well, that was before, now we've been shut down since last March. And it looks like we may be still unable to get back into the labs probably through the summer, and I think maybe till the end of the year. So we've been, have slowed down on getting students into the lab, but as soon as we get up and running again, I would love to offer different slots for students. And so I'm going to end with that. If there's any questions, I'd be happy to take them.
I do want to say that when the USDA was open before COVID, they also took our students on tours. So they would walk the students through different people's labs. It was a really great experience for our students and for our teachers. So the entire division that Jackie is working with, that Dr. Nesbit's working with, is beyond supportive of the SLT program and the biology and chemistry program.
Mm-hmm.
Anybody
Yes.
any questions? Great opportunity to talk to these researchers. Go, Aaron.
Okay, so you said between what age when they're infants?
Yeah, so the recommendation now is no at 4 months. So start introducing at 4 months, but it has to go directly in the mouth. If it gets on the skin, that's bad. So in the mouth, 4 months, no later than 6.
So the So it is genetic, but during that window, you have a unique opportunity.
I think there's a genetic aspect to it, but that I don't believe that has anything to do with the genetics. There is genetics involved, especially if you have asthma. So those are correlated. Asthma, dermatitis. Oh, Erin, I think you're cutting out.
Erin, you froze for a moment.
Yeah, I missed after asthma.
So that there is a genetic aspect to it. If you have allergy due to genetics, allergy, dermatitis, or I mean asthma, dermatitis, those are highly correlated with or food allergies in general.
That's fascinating.
Mm-hmm.
Does anyone else have a question for Dr. Nesbit while we have her on the line? Well, I just want to say, I just want to say that I've known you for, I think, 7 years, and I did not know that you were an accountant before all this.
I know it was. I was, because my big sister was, so I said, okay. And, you know, no offense to accounting, but I didn't like it. I'm much happier now.
Well, good.
But it did, you know, it paid the bills for a while.
Well, thank you so much for
Dr.
sharing, Dr. Nesbit.
Miss Amanda, just a
Any of
minute.
y'all are at Delgado, or no, she teaches at both. Oh, sorry, Habib, you want to ask a question?
Dr. Nesbit, just a very quick question. This is Dr. Ansari from Delgado.
Hi.
Is this USDA is nowadays completely closed? They don't allow for students or faculties to visit?
Right now, we are closed. We can, as researchers, request, and we can request permission to go in for maybe a week at a time for no more than 6 hours, but it has to be approved at an area level, so national level. And so, yeah, and nobody's allowed in the building other than that. It's shut down.
So we have to take the permission from USDA main office or from the director?
Yeah. No, from the main area office of the ARS, the Agricultural Research Service. Yeah.
Okay, I got you. Thank you.
You're welcome.
I have one more question.
Okay.
I'm curious about that. If you think that there's any possibility of peanut allergies and that window and maybe extrapolating it to other allergies like gluten or dairy, or if that window is just specific to peanuts. I don't know.
No, they have shown, well, peanut and tree nuts
right.
is exclusive. Yeah. Dairy and egg are kind of on a different spectrum because you tend to outgrow those allergies. So it's a little bit different. Gluten isn't really an allergy that involves IgE. It's not an IgE mediated allergy, which other food allergies are.
Okay, thanks.
Mm-hmm.
Thank you so much, Dr. Nesbit. I appreciate it.
All right, thank you. Bye, everyone.
Bye, y'all. Okay.
I will. Okay, got it.
So thank y'all for sticking around. Our next speaker is Dr. Webster Lewis, and he is the FST coordinator at Intertek. And I don't think you need sharing privileges, right, Dr. Lewis? Okay, perfect.
No, I don't.
Okay, thank you so much for your patience too. As you can tell, we're running just a little bit behind, y'all.
Okay, everybody. First off, I want to thank you all for having me. This is something that I definitely would have wanted to have when I was in school. Um, because the oil field and the oil industry, it's kind of closed off from inner city. I guess I would say youth. It's big in the river parishes, and a lot of people that I work with, they're actually from along the river, and they knew about it from their parents and their grandparents and stuff like that. But just a little about myself. I graduated from Xavier University in 2005. Um, I actually took physics at Delgado with Raymond Duplessis. He actually went to Xavier with me as well. Um, I went to nursing school for a couple of years, and I figured I'm like, I can't do all the blood and guts. So I took my biology degree and I have a minor in chemistry, and I'm like, I can try this oil field thing. I have a friend of mine that works for Marathon Petroleum in Garyville, Louisiana. And he was telling me about all the opportunities that you have in the oil field. So I found Intertek through a coworker who left the job that we were at, and he came over here and worked my way up. I started off as a lab technician, um, then a supervisor, and now I'm the FST slash laboratory coordinator. A little about Intertek. Um, we've been around for over 130 years. Um, we have 44,000 employees in 1,000 locations in over 100 countries. Um, the division I work in is the petroleum division. We have from New York Harbor to Los Angeles, we have analytical labs. Um, so now what I do, by me being an FST coordinator, I I coordinate with clients and our laboratory um, to let's see, for their testing purposes. We test crude oil, jet fuel. We actually test the jet fuel for Air Force One whenever it comes down. We do gasoline testing. So we have the FST coordinator. It's the freestanding testing. So this is testing that plants and even mom and pop oil companies, they have their own samples and they want us to test it and let them know, hey, um, is this crude oil worth the $10,000 that they want to sell it for? What's in it? The components in certain products that we test for. Um, uh, a company that I deal with, well, a plant I deal with is Valero St. Charles Refinery, and they do have some gasoline ships that they load, which is these ships dock on the river and they unload and load gasoline, and then they'll sell it to um, all over the country. But with the refinery, they refine gasoline and they pipeline it through throughout the country. So we take the final gasoline that they have and we basically certify it for them. Because it's like, hey, I have this gasoline and I can't just sell it to someone without a third party, a second party lab, which is what Intertek is, certifying it for them. Um, so we actually have in my lab now, we have 8 employees that work the main floor. We have 4 GC technicians, gas chromatography. We have 4 supervisors, and we have a lab manager and a branch manager. We have an operations side with inspectors, so they'll go out and they gauge gasoline ships, they gauge crude oil ships, and then they pull samples and they'll bring them to us for testing. So that's the IRT component of my laboratory, which is inspector related testing. As of now, we've actually, since the pandemic, we've had a hiring freeze. And it's been pretty tough for us. We have a lot of turnover at times because people move to refineries, or they just fall out of, the love of it. But it's a very lucrative field. It's very fast paced. If you like to always be working, it's very fast paced, and you can move around a lot. Like I said, we have labs from New York to California, and we do all types of testing. We have labs that test toys and the components in the toys, the plastics and stuff like that in the toys. So once you get in this company, you can move around a lot if you want to. Like I said, with my job, I coordinate. I was a lab technician. I still am to heart. I enjoy it. We have also we test octane readings. So at the gas pump, 87, 80, 89, and 93 octane reading. We test that for chemical companies like Valero and Phillips 66 and Chevron and Shell. We also have a cetane engine, which tests the diesel rating for diesel fuel. So we do testing on crude oil, like I said, crude oil, gasoline, diesel. And it's a field that if you have a 2-year degree from Delgado and you want to get a 4-year degree, this is the field that you can think about if you're in biology or chemistry. Actually, we've hired people with just a 2-year degree from Delgado. So that's an opportunity as well. At the moment, like I said, we're actually not hiring, but I've talked to April, and I also see Amanda on here, and I'll get her information as well. And when we do, I will definitely reach out to ones from Delgado or that's at Delgado, because we like to have people, that's passionate about the sciences, and it helps a lot in this industry and in this business, because it's more than just pressing a button. It's you want to know the science behind what you're doing. So when you do see results, you know if these are good or not to put out to a client. I really didn't have too much. I really would like to just answer questions if people had them. But anything about me and how I got to where I'm at, and okay, yeah, like I said, any people that how I got to where I'm at, how you can get your foot in the door. I really like to answer those questions. So I'll leave it open for the floor.
Dr. Lewis, this is Dr. Ansari from Delgado.
How you doing, sir?
Very nice presentation, and it is very encouraging for our students. So what is the possibility to visit your lab if you took our students?
Oh, we're open 24/7, 7 days a week. Anybody can come here. I get clients all the time. I get people all the time coming in, and they want to drop off samples and stuff like that. Our lab is located 149 Pintail Street. It's in St. Rose, Louisiana. And I see a question.
Thank you.
Oh, the lab that's located that tests for plastic composition for toys. That is our PCL lab, and it's actually located across the street from the petroleum lab that I work in. So we could walk over there as well if I had visitors.
They will be very interesting. You know, they will be get interested into the chemical field, petroleum field. So
yes.
we are planning to get our students over there.
Yes, and
Thank you very much.
I love to have them. You know, y'all can just send me an email and we can set something up. But like I said, it's a field that a lot of people don't think about when they're in the biology and chemistry and sciences when they're in school. And it's something that's been around a while. And like I said, people from the city don't really, it's kind of, you miss out. It's like, either I'm gonna be a teacher or I'm going to medical school. And you're like, I don't know about that.
Thank you very much. I can get the information from Dr. Amanda, and I can get in touch with you.
Okay, no problem.
Thank you, sir.
Kelsey, can you ask a question? Okay, yes, I'm sorry. Do you?
Yeah, I'm sorry. I was typing too fast. I'm sorry. But Mr. Lewis, I was just wondering, because you spoke about GCMS. Do you guys do testing for HPLC as well?
Yes.
Okay. Just wondering.
Mm-hmm.
All righty. Sorry about that, Mr. Amanda. I was typing too fast.
No problem. And actually, we just had a accident in our lab in 2019. So we basically got a brand new GC department. So if you guys are interested in gas chromatography and mass chromatography, it's brand new instruments, a lot of toys back there to play with.
Awesome. Thank you.
You're welcome.
Dr. Lewis, we have another question. Do you test Mardi Gras beads?
No, I haven't. I'm more than sure that we can test pretty much anything. Like I said, our former lab manager, Shanley Mullins, he's the PCL lab manager now. He's very a wealth of knowledge. He's been around a while, so I'm sure if we can figure it out, we can test it.
Hi, Dr. Lewis. Do y'all offer internships?
That we don't. I wish we did. Like I said, with COVID and everything, it hit the oil industry hard. And I did ask about that before I got on. No, we don't. We pretty much hire you straight on. So you wouldn't even need an internship. You get all the training from us at the lab. But no, we don't. Sorry about that.
If I can add something to that question, a lot of the interns, especially on the Chemtech side, they're usually hired and they just do their internship alongside their job that they already have. So if you did get hired, you could complete your internship even when you're still finishing up your degree with us.
Okay. And we also our last hiring we did, we got a lot of people from Tech 2000. So that's another avenue that we use to hire as well. Oh, we'll take all volunteers. We'll take all volunteers.
Does anyone else have any questions for Dr. Lewis? Well, we truly appreciate your time, Dr. Lewis. I have a feeling you will be receiving quite a few
No
emails.
problem. I give you all my email address. That's fine. It's Webster WEBSTER.Lewis, LEWIS, at intertek.com. And I try to help anybody with anything that they may have. Even if you're looking, as I do have some connections at plants as well. So even if you guys you see something on a job board and you're like, hey, I think I want to apply at Valero, I put in a word for you all, because I really would like to see people that, you know, from the New Orleans area get those jobs.
Well, I am sure that we will have a full crew of people taking advantage of that offer. The program is growing so quickly, so we're going to have more and more students prepared to go out into the workforce.
No problem. Thank you guys for having me.
Thank you so much, Dr. Lewis. I appreciate it.
All righty.
Thank you.
Bye-bye, y'all.
So our next speaker is Megan Morris. I'm sorry, I thought I took Adam Julian off, Megan, but Megan is coming from the AxoSim leadership team. And AxoSim was one of our earliest partners, and we are so grateful for their guidance and their support for the SLT program. So thank you, Megan. I am going to give you privileges to share. And there you go.
Absolutely. Thank you guys for having me speak. Let me pull up my screen real quick. Let's see. Oh, did it work?
We can see your yep, now we got it.
The full presentation? Full screen?
Yes.
Okay, awesome.
Yeah, you're good to go.
So good morning, everybody. Like Amanda said, my name is Megan Morris. I'm the Director of Operations here at AxoSim. We are located in New Orleans. And so today I was going to kind of tell you a little bit about what AxoSim is, what I do here, and then I'm really just going to tell you guys my sort of odd little story about my journey from high school to AxoSim and how I got here. I'd say I consider myself one of those cases of I want to be in the medical field, but I really don't want to be a doctor, paired with this non-traditional career path of I'm still figuring out what I want to be when I grow up. As I understand it, I think a lot of you may be in a associate's program, degree program, and have maybe some intentions to continue towards a bachelor's degree. So I kind of feel like maybe my story will strike a bit of a chord with some of you, just because you're still in the middle of your programs, trying to figure out, you know, where to go next. So what is AxoSim and what do I do? AxoSim is a neuro discovery service company, and what that really means is we help pharmaceutical partners, pharmaceutical companies, develop neurological drugs faster, safer, and cheaper by testing their compounds, so either drugs or parts of drugs, on our really powerful in vitro platforms. And so we can give them data that better predicts the data that they would see if they gave those compounds to actual humans, but we can do it in the lab. So we can use our two proprietary platforms called the NerveSim and the BrainSim to mimic the responses to drugs in humans. The NerveSim platform mimics the peripheral nervous system, and the BrainSim platform mimics the central nervous system. That's sort of it in a nutshell. And ultimately, we're helping the drug development process move along faster and cheaper. And what that translates to in real life is that companies can find cures and treatments to neurological diseases faster and for a lot less money out of the pockets of the people that are actually battling those diseases. And so what do I do here? As I said, I'm the Director of Operations, and so I do exactly what that sounds like. I direct the operations. I oversee all of the studies that we run here, and I manage all the people that execute those studies. It's a lot of hard work, but it's also really rewarding. Our company's mission is really easy to get behind because, I mean, who doesn't want to be part of finding safe treatments for things like ALS and multiple sclerosis, you know. But on the day-to-day side of things, being able to see my team, these folks in that picture, make breakthroughs and develop ways to generate really good, reliable data for our clients is pretty awesome to be a part of. Like I said, the folks in that picture you see, that's my team. I'm very proud to be able to lead these guys. Right now, it's a bit of an old picture just because clearly we can't, you know, huddle up and take a picture nowadays. But right now, the team consists of about 21 scientists, bioengineers, and support professionals of various types. Most of the folks that I oversee are research scientists or study directors or research associates and assistants. And that's probably what you're more used to hearing as like a lab technician. Our entry level job here is a research assistant. And those are the folks that are in the lab every day, pipettes in hand, growing cell cultures, running various assays on those cells with and without drugs. And from there, you can kind of be promoted through the research assistant levels depending on your proficiencies and your skills and how you sort of master those skills. And so how did I end up being the director of operations at AxoSim? It's a bit of a weird journey, so I'm going to back up for just a second. I came from a family full of medical professionals. My dad's a retired orthopedic surgeon. My mom was a nurse. My aunt was a dentist. My uncle was an OB/GYN, but I did not get that gene. I, like Dr. Lewis, was like absolutely no to the blood and guts. But I love biology and I love science and I love the idea of the medical field. But the one thing I knew I didn't want to be was a doctor or a nurse. And so growing up, all I really knew was that I like to build things. I like to create things. I like to take things apart, figure out how they work, fix them, that kind of thing. And in high school, what that translated to me was be an engineer. And I took, I'm sure, all the same aptitude tests that you guys have took during high school, maybe even when you got into college again, to try to figure out what you want to do. And what I ended up deciding on was that I wanted to go to college to be a mechanical engineer. And I had rolled in the program at LSU when I was packed and ready to go. And a couple weeks before I moved, I got some pamphlet in the mail, I think it was from LA Tech, about biomedical engineering. And I was like, what is this? I could marry biology and engineering and actually get a job doing it. So I switched majors. I switched to biological and agricultural engineering at LSU. And when I was there, I got a student worker job at Pennington Biomedical Research Center. And when I was there, I learned the basics of research, some special assays, a lot of what the core is of what the lab folks do here for us as well. But what I really got to see was this interaction between researchers and what it was like to work with them. And what I saw was a lot of collaboration among people all trying to find solutions to problems. And that was really attractive to me. So I graduated with my bachelor's, but I still wanted to pursue the medical path of engineering. And so I decided to stay local and applied to Tulane's biomedical engineering PhD program. And Hurricane Katrina hit in the fall of 2005, and that was supposed to be my first semester there, which clearly did not happen. The university shut down for the semester, and so ultimately I started the following spring. About a year later, I came to this sort of personal realization that I really didn't want to be a professor, and I probably didn't want to be a traditional researcher. So a PhD program was maybe not the right route for me. So I transitioned to their master's degree program and graduated in 2008. And that's when I got my first big kid job. And I'd say that's where the real learning started. So that first job out of grad school was with a company called the Bioengineering Group. And contrary to what the name might imply, this job had, at first, what I thought was nothing to do with biology. We worked with the Army Corps of Engineers to design all the new levees in and around New Orleans after Hurricane Katrina. And it was about 3 years later when we were pretty much done redesigning all the levees and flood walls. And after a lot of them had been repaired or rebuilt, when I realized that this job did have a lot to do with biology. It was just the perspective that I had to look at it through. Every person's life that we saved by protecting their homes from the next flood, every animal's home that we avoided disturbing to build that levee or that flood wall, what I learned was that my perception of what my career could be was even less black and white than I already thought it was. And so I went on to spend about 7 years at a company that had even less to do with biology. I worked for a company called Global X-Ray and Testing Company, which interestingly enough was purchased by Intertek. So that's where Dr. Lewis works right now. And anyway, they're a non-destructive testing company, which basically meant they performed types of quality tests on pipe welds. So it was very much oil and gas industry. And honestly, I took that job because I needed one. But you can see that I started this sort of path of managing different aspects of the same business. And so what these jobs did for me was to provide me with an opportunity to learn how a business worked from the inside out. And I was able to dive into a lot of details of a lot of different departments and develop processes that improve the way that business worked. I'm intensely grateful for my experiences with these companies, but I was still looking for that connection to biology, to life, right? And so every now and then I would do the whole job search thing, but honestly, I didn't even know what I was looking for. I felt like I had been away from traditional biology for so long that I wouldn't be able to get back in. And I felt like I had gotten this fancy engineering degree, but I really wasn't an engineer. And so I started to think, what skills have my seemingly random jobs given me? Well, what have I learned from my past experiences that could translate to any job? And in my effort to find those answers, I not only found AxoSim, but I also figured out that I really was an engineer. At my core, through all of those jobs, what I was doing was collecting and absorbing and analyzing information in order to creatively and systematically come up with solutions to problems. And at its core, that's exactly what engineering is. One day I was laying on my couch scrolling through Indeed, and I found a job posting for AxoSim, and the rest is sort of history. Now I use all the things I learned from those previous experiences and the education that came before that to oversee the operations of a biotech company with some really cool and powerful technology. My biggest take-home message here is that if you don't exactly know what you want to do with your degree and you haven't figured it out yet and you still don't know what you want to be when you grow up, it's totally okay. Most of us are still figuring it out every day. But the most helpful thing I think you can do for yourself is to look at the things that you learn how to do, look at them with a different light. Ask yourself, what did I really just learn and how can I apply it elsewhere? And when you figure that out, like, the whole world is open to you. And so as you could see, I had a handful of jobs, but if I've only learned one thing at all, it's that I have so much more learning to go. I wanted to mention the two things, excuse me, that helped me tremendously, not only during college, but that are still probably the most helpful takeaways for me every single day that I'm here. And the first thing is learn how to learn. I'm not saying don't study, but in the act of studying, try to figure out how to figure out the answers to the things that you don't know, and not just by Googling them, right? Really figure out, how do I answer questions for myself? And the second thing, and probably the biggest thing, is about the attitude that you bring to school and to your job every day. Be open to knowledge, be open to other people's experiences, be open to opportunities, even if they don't seem like they fit your life plan. Just be open to them. They may be something really amazing. Be absorbent of the information that's thrown at you. Don't be afraid to ask questions, especially things that don't make sense to you. Be curious. Always be curious. Ask questions, and asking them is really powerful. And last thing is stay humble. There will never, ever come a day where you know it all. I promise. Even when you're an expert at something, there is always someone somewhere that you can learn a little bit more from. So be open, absorbent, curious, and always humble. And so, yeah, thank you, Saul, for having me, letting me ramble. I put my LinkedIn profile name up there. That's really the best way to reach out to me in a networking capacity. And also, I have our website. I really encourage all of you to visit that website. We have some great information, some really cool videos that sort of explain better than I could ever explain in 15 minutes how our technology works, as well as some publications that we've been a part of. And that's kind of it. So I guess if anybody has any questions, you guys can throw them at me.
I want to say that you've inspired me, so thank you. I don't think that a lot of people realize the paths that we take because we're in a position. So yeah, we got there, but the journey was part of the process.
Absolutely.
I actually had one question, Ms. Morris. I wanted to ask you about your student worker position at Pennington Biomedical Research Center, because I was actually thinking about doing that, but I know right now with COVID and all of that, everything is harder now for stuff like that. But I was wondering, did you actually, like, was it beneficial? And was it in your actual field, or were you just a research student in another field?
It was tremendously beneficial, and I probably will say, as odd as that job was, it still kind of remains like my favorite job. So I worked for a lab, and we, I don't remember exactly what we were testing because it was so long ago, but it was all revolving around fat tissue. And so I managed the mice colonies. I was able to run, different DNA panels, PCRs on those mice, and I worked my way up into a research associate position before I left there. So it was all revolving around biology in some way, shape, or form. And I think at that point in time, I probably really didn't even know what my field was. So for me, it was always a yes. It's part of my field. But yeah, that job was amazing, and I think probably paved my way to stay in biology.
Okay, good. Because I was thinking about doing that as my first step of while I'm in school, or even after school. I was thinking about Pennington Biomedical, because I'm kind of like on the limb of what you were saying. You have this degree, or you're about to finish and get your degree, but you're really like just stuck on what exactly you will do. And that's kind of where I'm at, but I looked into Pennington Biomedical, so that's why I was wondering.
It's a great place. I would highly encourage it.
Okay, thank you. And I actually, I'm about to add you on LinkedIn as well, if you don't mind.
No, not a problem. Go for it.
Thank you.
Anybody else have questions for Ms. Morris while we have her on?
Hey, can you hear me?
Yeah.
That was an amazing presentation and very encouraging to me too. I was wondering if you could give a quick rundown about what those technologies are, the nerve sim and the brain sim, like just a little more detail about what they involve.
So both of them are just different platforms. And so by platform, I mean they're cells grown on a different substrate, right? At its simplest explanation. And so the nerve sim is some peripheral nerve cells. We have a few animal models and a human model. And then the brain sim is central nervous system cells grown together in a culture so that it mimics the relationship between those cells that is similar to the way it works in the body, but obviously maybe not quite as complicated as the way it works in the body. And so what we'll do is take, our clients, the pharmaceutical partners, their drugs, put them on those cells and then test them in various ways to see what the effects are. Sometimes it's looking for the efficacy of that drug. Does it increase some effect, or is it harmful to those cells? It just depends on what that client is looking for, but that's sort of it in a nutshell. We apply drugs to cells and see what happens.
I have a question. Can you hear me?
Yes.
I was wondering what is required to obtain a job at AxoSim. Would the associate's degree from Delgado be a good starting place, a minimum requirement? I have graduate degrees in other fields, and I'm going back to school. And I was wondering, should I apply after finishing a degree or while I'm doing this degree? Or does it require, say, a master's degree from Tulane Biomed?
No, not at all. We have people with, I don't think we have anybody with an associate's degree. However, I would not close the door to that. We definitely have many people here with just bachelor's degrees. And what we're really looking for is probably more than the degree you have is the experience that you come with. And so to start out as a lab technician, what we're really looking for is this really good core understanding of biology and cell culture. And a lot of times, if a person does not have some sort of base cell culture experience, that's sort of where that dividing line is where we move somebody to the interview stage versus, unfortunately, not. That's sort of that core skill that we're looking for a lab technician versus, like the lab that Dr. Lewis ran. It's a different type of testing and a different type of cells, and so understand what you're testing is really important here. It's a very fast-paced environment once the people get hired, and so we train from bottom to top, but you really have to have that sort of base understanding to start here.
Do you have internships?
We do right now. It's a little funny because of COVID. We have a couple of virtual interns that we're doing through the Youth Force NOLA right now, but that's all we're tackling in the midst of COVID. But I'm hoping next summer that changes.
Okay, you said it's Youth Force. However, I'm rather old, and what opportunities are because I've run into a wall there with a lot of education in my age, and I can't get entry levels anywhere. So
yeah, I don't. That would definitely not disqualify you at all from here. It's just that the internships that we're able to offer right now are sort of limited just because of the physical ability to be in the lab. We've sort of cut that off during COVID. Just to limit the safety of our employees. So right now it's just these virtual internships, and the connection that we made to get those virtual internships was through Youth Force NOLA. That's not our only method of having interns. It's just that, like I said, during COVID, the need to have people physically here was sort of, unfortunately, not available.
Thank you very much.
So I just want to emphasize that AxoSim has supported our students pre-COVID in the internship program. They have to go through the process of applying for the internship, interviewing for it. So it's a great door and experience to open. But even more exciting is after Ms. Morris, we actually have one of the co-founders of AxoSim going to speak to y'all about the company and other opportunities and experiences. So it's definitely something to stick around for. And Megan, you really have been so inspirational. Thank you.
No problem. Thank you. I'm gonna actually stick around for Dr. Moore's presentation, if that's okay.
Oh, absolutely. And Dr. Moore, do you need me to give you sharing privileges?
Yeah, I think I will show a few slides. I'll try to not be in professor mode and show 45 slides for a 15-minute presentation. But I probably will show a few slides to try to give people some context. I really appreciate Megan's approach to what she shared, because I think it's so important to know that very rarely do people in their teens or 20s or even later in life know exactly what they're going to do, long term for their career. It wasn't that much different for me. I grew up in a little bitty town in western Nebraska with one stoplight. And so, growing up in that town, anyone who did well in school, everybody told you that you should be a doctor. And a lot like Megan, well, I didn't have a problem with the blood and guts, but maybe I just rebelled against that idea that I needed to be a doctor. And so I just decided to do something else. But I didn't know what I wanted to do. I went into engineering, didn't really know what that even meant, coming from such a small town, didn't know what engineers were, what they did. So anyway, all of us have a story like this, which is, we didn't have this idea in our heads that we wanted to do this one thing, and we just did everything we needed to do to get to that one goal. It almost never works that way. And so I found Megan's presentation to be super encouraging for that reason. And maybe you all did too. So I wanted to share maybe just a little bit more about what AxoSim does from the technology side. I hope people can see my screen. Is that correct?
We can.
Okay, so I have two different presentations. This started many years ago in my laboratory. We were working on trying to find a different way to model the peripheral nerve. Okay, so a lot of you might not know too much about this, but everybody knows where your brain is and your spinal cord. Okay, and then the peripheral nerves are all the nerves that allow you to move your fingers and toes and feel what's going on, your hands, your feet, your trunk. All that information is taken from peripheral nerve and it goes to the spinal cord, and then that information eventually goes to the brain. So the peripheral nerve is made of your sensory nerves and your motor nerves. The sensory nerves allow you to feel everything. The motor nerves allow you to move everything. And then a lot of us forget about the autonomic nerves, and those are nerves that go to organs like your heart and your gut and so forth to make those work properly. So some of you might know people who are, for example, taking chemotherapy, and they're trying to cure cancer with these drugs that we know are poisonous, but the hope is that the poison kills the cancer before it kills anything else. And that works some of the time, maybe even most of the time. But the problem is a lot of those drugs cause a lot of toxic effects to your peripheral nerves. So some of you that might have family members or friends who are on chemotherapy, you may have heard them complain about tingling or sometimes burning sensations in their hands and feet. This is a very common side effect of chemotherapy. And the reason is because your peripheral nerves are made of cells that have their nucleus near the spinal cord, but then they have to project long distances, from your neck, down your shoulder, down into your arm, and all the way to your fingertips. Those are cells that have their nucleus near the spinal cord, and then the tips of them go all the way down out to your arms and your hands and your feet, so forth. And so because there's such long distances that cells have to, they have to traffic the enzymes and the machines and other things that make the cell work. They have to do that over a long distance. And so that means that your nerves are very susceptible to toxic effects of drugs like cancer drugs. So what we did here at AxoSim, and what we started in my lab, is we started working on this model that I'll show you this little video to give you an idea of it. And so what we do is we culture cells. So Megan talked about this a little bit. So we take cells, sometimes we take them from rats and mice, sometimes we take them from, we derive them from stem cells that were originally taken from humans. And then we make a little spheroid out of those cells, and that spheroid is like less than 0.5 millimeter in diameter, so really small. And we put it in this gel that has a certain shape to it. And so when those parts of the cell called axons that grow those long distances, when those start to grow out from the spheroid, they're forced to grow down this little channel. And that channel is really important because that is what allows us to get a very specific structure of the tissue that allows us to get these very specific measurements that we take using electrophysiology. We literally zap the cells with a little bit of electricity, and we measure how fast it goes down the cell, and that tells us a lot about the health of those cells. But then we can fix that tissue and slice it and take pictures under very powerful microscopes, and we can get even more detailed view of the health and well-being of those cells. So just to give you a little some colorful pictures of what this looks like, these are what the cells after they're made into a spheroid look like under a microscope. This is kind of a cross section showing the spheroid has this three-dimensional structure. I think Megan showed this picture earlier, the cells growing down that channel. We took some special kind of microscope images where the different colors here represent cells that are growing in different depths. And then, as the video mentioned, we put electrodes in there and we get these population responses that have a certain shape, and we can study the shape and how fast the population responses, what they look like and how fast they go, and we can take detailed electron microscope images in order to look at, again, the health of those cells. So all of this is to say that in order for a person to know how to do all of this kind of stuff, it takes years and years and years of schooling. So I went to school, I got a bachelor's degree in engineering, and then I went and got a PhD in biomedical engineering. And then I made, and all of that took about 10 years. And then I spent another couple of years learning how to do research as an independent researcher. We call that a postdoc. So I did a postdoc for a couple of more years. So it took me probably about 12 years of schooling and training to learn how to do all of that. But the good news is, because let's face it, not everyone likes to be in school for 10 to 12 years, right? In fact, it's probably safe to say very few of us want to be in school for 10 to 12 years. I'm probably an anomaly. I'm just one of those people that just love school. And in fact, I never left. I'm now a professor at Tulane. And so I sort of just stayed in school and never left. But the good news is, in order to all of those aspects, right? So we have students in my lab here at Tulane who are training to be scientists. And so, yeah, they stay in school for a long time, and they learn all of the, they learn how to do, they take all the classes, but then they learn how to do most of the stuff kind of on the job training. So the PhD is almost like one of the last apprenticeships, because they spend a good 4 or 5, sometimes 6 years just in the lab learning how to do research, learning how to be a scientist. But the interesting thing is, that's what happens in my academic lab. But at AxoSim, where we're trying to do this for as a commercial entity, all of that stuff that I showed you has to be done much faster, on a much larger scale, and much more efficiently. And so one of the ways to do that more efficiently is that not every single person has to know every single aspect of everything that's going on. There's a lot of stuff that I showed you. I threw at you really quick, right? From microscopy, anatomy, physiology, cell culture, and you have to have a really deep understanding of all of that kind of stuff in order to invent it. But you don't have to know every part or of every aspect of everything in order to contribute. So like Megan said, at AxoSim, we do have scientists that kind of have this big picture. They kind of know everything that's going on. But we also have laboratory technicians, laboratory assistants that, you really only have to know a certain part. So we have lab techs that culture the cells, and they might take those cells out of the incubator and go take a picture on the microscope. They don't have to know all that much about anatomy and physiology and engineering and everything else. And so for people who don't want to be in school for 10 to 12 years, like I did, we also have jobs available. And that's one of the reasons that we're really supporting this program here, because I do think cell culture is one of those skills that are going to be needed more and more in our society. You think about these vaccines that are coming out right now. In many cases, those had to be tested on cells before they were injected into humans, right? And so somebody had to do the hard work of culturing those cells, putting the vaccine on those cells, et cetera. So I do think there's a lot of opportunities, even here in New Orleans, for students with associate degrees, because just like Megan said, it's really the training that is more important than the credential. Okay, so we are able to, like she said, I don't think we have anyone yet from with an associate's degree, but if somebody had an associate's degree and they were really, really skilled at cell culture, that would mean more than somebody with a bachelor's degree in biology who had never done cell culture, right? And so I think that's hopefully helpful for you all to know. The last thing I'll say is, even though you may not have to know everything about everything that's going on, you still have to be a highly skilled, highly technical, and very careful individual. Some of the things that we work on, one mistake can be very, very, very costly. And I don't say that to scare anybody. I mean, you know, mistakes are just that. They're mistakes. They happen. We live with it. But if a person is prone to making mistakes again and again, and they're because of sloppiness, it's not gonna work. That's gonna be a recipe for disaster. It's gonna cost the company a lot of money. It's gonna cost the team a lot of time, because other people, you know, contribute. And so if you're sloppy and you don't take good notes, you don't take good records, or you're not very skilled with the work that you're doing, that's not going to be acceptable. And so it's kind of an interesting time where there's a great need for skilled individuals like to do really highly technical work like cell culture, but that may not require 10 to 12 years of schooling. But like I said, it's still very important that each person takes responsibility and learns the job and learns it really well, and is able to be meticulous about the work. And so I've probably talked enough already. I can entertain if anybody has additional questions that Megan didn't already answer.
Dr. Moore, I have a question for you. This is just a pure of interest question about the science that you guys are leading with at AxoSim. And I know that there's been some recent studies and publications about these mini brain type structures, which are more composites of different neurons and different types of cells that make up the brain. Is your technology adaptable to some type of emulation or modeling for those mini brains?
Yeah, so that work didn't come out of my lab, but AxoSim has partnered with a professor at Johns Hopkins University, and we've licensed technology from them to exactly culture those mini brains. And so we are in the process of we've got them in the incubator right now, and yeah, we're growing those mini brains. One of the difficulties with those is the way most people do it. They kind of just let them grow, and they keep growing, and they keep getting bigger and bigger, and at some point, it's not very reproducible. So you know, if you make 100 mini brains, you know, they're not very similar to each other. So one of the reasons that we licensed the technology out of Johns Hopkins is because they have a very specific method that allows them to be much more reproducible. The size is more consistent, and also the composition of the little organoids is more consistent. And so we thought that for a company that needs to do this on a larger scale and do it again and again and again with high efficiency, we thought that this technology was more attractive.
Thank you.
Yeah, you bet. Thanks for the question. I think you're muted, Amanda, if you were saying something.
We need to have a bingo card for Zoom.
Indeed.
Did anyone else have a question for Dr. Moore?
Yes, a quick question. Would you be willing to meet with Delgado students just to discuss this further at some point, or is it too difficult because of COVID and you have too many other students?
Oh yeah, I always forget about COVID. Yeah, you know, I'm certainly willing. There's always a way. So you can feel free to reach out. I think Amanda's providing my contact information. Feel free to reach out. I'm not always. I'll just apologize in advance that there are certain times throughout the semester where I just get so behind on email that, you know, I might not be the super fastest to respond, but I do my best to respond to every every single email that I get. Well, unless it's spam. So I think if you just introduce yourself, remind me in that email that we met at this event, I will do my best to get back to you. And I'm more than happy to talk with folks who are interested in this kind of thing.
Thank you.
And I teach the cell culture class at Delgado. So if there are any specific techniques or if there's anything that you think needs to be really addressed with our students or would be helpful for our students to have an understanding of, please reach out to me and let me know. And I'm happy to kind of incorporate new techniques into our course as well.
Yeah, how wonderful. I mean, this is really a great resource. I've not heard of a program like this before, and that's why we're such strong supporters. I think there's just really an excellent opportunity here to capitalize on this. So yes, Dr. Deku, we should brainstorm about that. I think one thing we could do is you could even come either to the company or to my lab, and we could, you could just observe the way we're doing things. You know, cell culture is one of those things where everyone thinks that everyone else's method is the wrong method. I still have yet to figure out what the correct method is. But everyone thinks that everyone else's cell culture technique is wrong, which is so funny.
One of the students was bright enough to say, well, you do it exactly how your boss says to do it.
Yeah, that's the way you do it. That's the right way.
That's exactly right.
That's the right way for sure. Yeah, we should brainstorm about that.
Yeah, sounds good.
Likewise, feel free to reach out to me. You could drop by my lab or the company, whichever I think makes the most sense, and we might be able to come up with some ideas about best practices or how to new things you could incorporate into your course.
Okay, well, I'll reach out sometime in the next week.
That'd be great.
Yeah, you can certainly reach out to me as well on that topic, because I find the one thing that's often missing is the component of touching or working with human cell lines. It's often very different than working with animal cell lines or bacterial cell lines or something like that. So,
and unfortunately, we do not have the ability to work with human cell lines right now at Delgado. And I mean, I've worked with A549s or other human-derived cell lines, but currently we can't do that. But I could definitely, touch on that in a lecture component.
Absolutely. I think we can get creative with something.
Yeah. All right. Well, I hope to talk to you guys soon.
Great.
Thanks, guys.
Yeah. All right. Thanks a lot, everyone. Thanks for having me.
Thank you, Dr. Moore. Thank you, Megan. We really appreciate your support. And Charlie and I are just texting saying how proud we are of everybody and everything going on. So thank you.
Thank you.
Yeah, wonderful. Thanks a lot.
Bye.
So I am excited to introduce our next speaker. We'll go in a little bit out of order, but our next speaker has been also a supporter of Delgado's SLT program since early on, since pretty much the inception of the program. And even more so, this individual, Dr. Tubon, comes to New Orleans and hosts NSF workshops for our students, doing everything from stem cell culturing to CRISPR. So we're very fortunate to have him and his resources as part of the Delgado support team. So Dr. Tubon, do you need sharing access?
Yes, please.
And I apologize, I have to let this little critter outside.
No problem. So this is a great transition to follow up with the presentations we just heard from AxoSim. One of the areas that I've been very, very interested in is developing programs for stem cells and regenerative medicine. And as Dr. Rosenzweig made a note of, I've been down to Delgado Community College and worked with Dr. Deku and Ms. Shunick on several occasions to help develop some of that programming, and we're going to continue to do that. So if you would just allow me one moment, I will share my screen here with you, and I'm going to go over some of the things that I'm involved with that really are crossovers with the program at Delgado Community College. All right, so I think Dr. Rosenzweig, you need to stop sharing that screen. There you go, and then I can. Thank you. So as Dr. Rosenzweig mentioned, I'm with Madison Area Technical College, and so I'm about 1,000 miles north of where you guys are at. It's a little bit colder here, but not that much colder, at least not in recent days. So it is a pleasure for me to be here, and I thank you for the invitation to come and speak. By way of introductions, so I am a professor in the biotechnology department at Madison Area Technical College. We are a two-year community college. We were one of the first programs to start biotech back in 1987, and since then, we've seen the development over 400 different programs across the United States. And so as part of that, we've been part of the National Science Foundation efforts that are funded to develop programs in biotechnology, which brings me to work with Dr. Rosenzweig and Ms. Chinook and the crew down at Delgado on programs and bringing in any resources that we can from the national infrastructure. So my background, I'm originally from San Diego. I am not a Midwest person. But many years ago, I made the jump from San Diego to New York for graduate school. And in that capacity, I studied cancer biology. Did that for my graduate work and then jumped ship very quickly after that and landed somewhere in the Midwest in Madison, where I pursued my postdoctoral studies in neuroscience and in stem cells and regenerative medicine. And then shortly after that, I took a position at the University of Wisconsin-Madison, and then made a transition from there and leap of faith into the two-year community college to help develop programs for workforce development. So my image here is just a little bit of a reminder to say that no one's career is actually straightforward. There are a lot of directions and turns that people will take, but ultimately, if you find yourself in a position where you love what you do, you can't go wrong. So, find those things in life that really make you happy, that drive you, that are those forces which really motivate you to get up in the morning and say, hey, I'm ready to go into work this morning. So in any event, my current position is at Madison College, but I also hold several other positions actually with the University of Wisconsin-Madison in their Wisconsin Institutes for Medical Research. I'm one of the co-PIs on our National Center for Biotechnology Education, which is called Innovate Bio. And I run a number of grants, including a coordination network for cell tissue manufacturing and part of something called the ERA Center, which is looking to really build the capacity for workforce and looking at the impact on the community. And I work with a number of our research centers, including our engineering research centers as well. So my career pathway has taken me from undergraduate work in general biology to graduate work into a number of different fields. And geez, I can remember a time when I would spend late nights working on ground squirrels. And I went from working on ground squirrels, which is a really interesting model, to working in fruit flies and then working with stem cells. And you ask yourself, with all of these different models that are out there, we're talking about just biologic models to study living systems. With all of these different models, what is the connection here? And really the connection is pretty clear. What's clear at the time? I was asking the same question in all those systems, and that is, what is the pathogenesis of certain neurological diseases? So regardless of what that system is, there's always more ways that you can try to find that answer. And that's one of the beautiful things about science and what this field is all about. So with workforce careers and what we're doing here, I wanted to make one important point, and that is, these workforce programs, developing programs for biotech, developing programs to get people ready to work in companies like AxoSim, you should kind of get this good sense that this data is really important. So when we originally surveyed a host of colleges and universities and their chief academic officers and asked them if their students are prepared to enter into the workforce, 96% of them said yes, absolutely. Our students are getting the right education that they need to get into those jobs. Well, we flipped that around and we asked the same question to employers. And what we found was that there's a serious misalignment. So 11% of those employers were saying, yeah, there's no matchup here. Those students who are coming through the education system are well-rounded, they're well-informed, but yet they don't have those skills that are necessary to help us do what we need to do, especially when we talk about complex companies and jobs and skills and competencies. And so I jumped headfirst into that area to see what we can do, and through the Community Tech College, develop those kinds of programs. So long story short, this is a little bit of a summary slide that was put out by the National Science Board. And it's just here to remind us that there's a prediction of about 3.4 million open jobs out there that represent those jobs that are going unfilled in the middle skill work set. So you're asking yourself, what is that, right? Well, the middle skill work set they're the folks who have maybe more than a high school degree, but not yet have attained their bachelor's degree, or maybe they have a bachelor's degree, but they're coming back into the community college to get retrained and ready for the workforce in a very specific field. So 3.4 million is quite a number of jobs. In the next 5 years or so, that number jumps up considerably to all of STEM to about 25 million from projections out there. So programs like the ones at Delgado Community College are really making headway with changing the message of what these kinds of programs are doing to help grow our workforce, focusing on data, making those partnerships happen, and then working across institutions to make sure that students get the experience that they need, which is what the beautiful thing is about the biotech program. So our partnerships with Delgado, we've had numerous partnerships and we're going to continue to develop those over the next few years. So as part of our national center, we have about 136 community colleges across the United States that we oversee, or we provide or help them with resources for their biotechnology programs. And for what that means for us, what that means is really developing activities like the ones that we support with Delgado. I've been down there several times over the past few years to help develop workshops for students and faculty in stem cells and regenerative medicine for workforce skills. We've done genomic approaches, gene editing, and CRISPR technologies. We've worked with Dr. Rosenzweig and Ms. Schunick on cross-institutional networking to support meetings and conferences. And we had a number of students from Delgado last year that we've invited to attend conferences with us. And we're going to continue that partnership with you all as well. So there's a number of ways that we can continue to do this, but I think that the most important thing that I want to bring up, and I'm going to fast forward through this. I want to make one really quick stop here. These are images of our students and of the folks down at Delgado. Some of them you'll recognize, like Ms. Schunick here all the way on the right. You know, we'll take students down to visit with partner colleges and do industry tours, and then we open up the doors to both ways in order to get that national sense of what's happening in the world of biotech. Okay, so I'm interested in core skills and abilities, as everyone is, but making sure that when we develop these programs, those core skills and abilities translate to meaningful jobs and then meaningful applications in the workplace. And so I'm not going to go through all of this stuff. I think that faculty did a great job going through the core program, but these are all things that we're addressing here. Biomanufacturing knowledge and skills and abilities, documentation. So let me make one comment about this slide. I'm not going to go through this. This is about 15 different competencies for biotech workforce. Each one of these competencies is embedded in the instruction for the biotechnology programs. And that is why these biotechnology programs are developed specifically to address industry and also to provide additional opportunities as students transition to other additional degrees, like a bachelor's degree. It's a win-win situation, folks. And I'm glad to be a part of the whole the whole ecosystem that's developing this. So I think I might be running out of time here. I know we're pushing a little bit on the time scheduling, and I wanted to leave some questions for you guys to ask about anything at all, not only about biotech programs, but about, sorry, I'm gonna fast forward to this really quick, but about programs from across the United States. And so Dr. Rosenzweig and her colleagues and the team with the biotechnology program are part of our Innovate Bio Center, and we are proud to have you all as partners in our efforts to grow biotech. So back to you guys.
So does anyone have any questions for Dr. Tubon? And while he was saying biotechnology, when he comes down, it is a collaboration between biotech and chemtech students and faculty. So you always have the opportunities to learn skills within both concentrations.
Tubon, this is Habib. When are you visiting us next time?
I'm sorry, it's good to see you, Dr. Atari.
When are you visiting next?
Well, it's a very interesting question. I would say as soon as it's reasonably possible and it's safe to travel. You guys have an incredible industry down there in Louisiana, in the New Orleans area. There's a lot of growth potential for biotech. You guys have New Orleans Biotech Innovation Center, multiple partners down there that we are working with on a regular basis. So it's definitely a good reason to come on down and see all of you guys, but also to know what's going on there with the developing industry. So as soon as reasonably possible.
Good to see you. We are excited to see you.
Thanks, Adi.
Thanks.
Well, we appreciate you spending the time with us today, Dr. Tubon, and sharing your knowledge. And again, y'all, once COVID restrictions have been reduced, there are so many opportunities that both students and faculty can participate in. And then as we close out today, one of our speakers is also still in graduate school, so they were not able to attend due to some unexpected occurrences that came to surface. But what I want to make sure is that everyone, before we let you go, you understand that the relationships and partnerships that are being built within Delgado and the community is immense. These are just some of them, and these cover both chemtech and biotech. Here are some of our national relationships that we have. Most of these are through NSF, Department of Energy, Department of Defense grants. So again, lots of opportunities for students to go to different conferences, to present, or to just learn. In the chat, I put a handout. The handout is for y'all to be able to meet our team and to have a little insight to some students' experiences. And I've also put the speaker handout where it has the introduction to all the speakers. This is just an overview of where some of our students are at now. And again, these students are either going straight to the workforce, or they are going back to school to pursue a bachelor's, master's, and maybe even a PhD. Again, most of our students are earning between $42,000 and $62,000, which is pretty outstanding. And again, we just want to talk about the growth of the program, and this is only possible because of our partners and because of students like you. We are started off in 2017 with 4 graduates, and we have grown by a few 100%, and we are expecting to graduate 7 to 9 this year. So we are doing very well, but we can always strive for better. And last, again, I want to thank you for your time, for your questions, for your participation. And I'm excited to introduce Dr. April Noble. She is the Director of Chemical Technology, and she is just going to close us out for today. April, do you want me to let you share screen?
Oh no, I didn't have anything. Yeah, I was just coming in to pop in.
Okay, well, April is Chemtech director, and she teaches the higher level chemistry courses, which I cannot speak to because I did not do well in chemistry. But again, if any of you ever have questions, let me get up there. You can email any of us. Sorry, little bit happy mouse. You can email any of us at these emails, and any questions you have, there is no question that is a stupid question. So if you have any question, any information you would like to answer, please reach out to any of us. Charlie, do you have anything to say, or Deandra, do you want to close out?
I just wanted to say thank you so much for coming, and thank you to all of our speakers for giving a real world perspective to our students so that they kind of understand that your goals don't always land you exactly where you think you're going to be. That doesn't mean that you're not going to be successful and happy with the career you end up in. And I fully believe that our program does set you up for success in the future, no matter what you want to do. So we're going to be here to support you guys. Feel free to contact me anytime. And, you know, I'm on your team. So thank you so much, everybody.
Hi, I want to say thank you as well to our presenters and to the participants for spending your time with us today. We do hope to do this again in the future. Hopefully after COVID times, we can see y'all in person. This was a great presentation via Zoom, but I know it could be so much better in person. I do want to make sure that the participants know that they can contact us at any time. They do not have to go to the actual advisors. We will be your personal advisors for this program. So be sure to contact us, send us emails at any time. We will help you with whatever you need, whatever we can. We are friendly. Don't think of us as just these professors. We're real people. Megan did a wonderful job of explaining, and so did Webster Lewis. He also did a good job at letting you know that this is real life. You may not know right now what you want to be when you grow up, what you want to do for the rest of your life. But we want to present this as an option for you. We at least want you to come in, take an intro to science technology course, and you may find even more that you're interested. But it is a very good option. The jobs provide a viable living wage. So if you have any other questions, just let us know. We're here to help. Thank you.
Thank you, everyone. This recording will be posted onto the website as well as the handouts. And again, we really appreciate your time. We appreciate the speakers' times. And as Deandra and Ms. Schoenig said, reach out to us. We are very approachable. Thank you.